Okay, it's 8:33 and it looks like the number of people joining has slowed down. So let's go ahead and get started. Good morning. Welcome to the Harmful Algal Bloom Research Symposium. My name is Amy Weckel and I'm from the Illinois Water Resources Center at the University of Illinois at Urbana-Champaign. I'm joined today by Eugene Bragg from Ohio State University Extension, and we will be your moderators for this morning's session. The Algal Bloom Action Team is thrilled to be hosting this event for a second year, and we are so glad you're able to join us. And there we go. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resource Research Institutes, the North Central Region Water Network, and university extensions within each state in the North Central region. In addition to hosting this annual symposium, our team is developing a website of resources that can be used freely for outreach and education around harmful algal blooms in the North Central region. I will drop our website in the chat in a few minutes, and I encourage each of you to visit that website. There you can explore some of the resources we have to date, including a What You Should Know fact sheet, a HAB's Frequently Asked Questions database, and recordings from our bi-monthly webinar series, as well as last year's virtual HAB's Research Symposium. Thank you, Anne. She just added our website to the chat, so you can access it there. This year's symposium is organized into 4 sessions over 2 days. Today's sessions focus on HAB's monitoring and ecology in the morning and cyanotoxin treatment and detection in the afternoon. Friday sessions will focus on forecasting and modeling HABs in the morning and emerging technologies for detection and monitoring in the afternoon. Each session will be facilitated by members of the Algal Bloom Action Team. Before we get started, just a few housekeeping items. We will have 4 30-minute presentations this morning with a 15-minute break between the second and third talks. At the conclusion of the 4th talk, we will hold a 30-minute presenter Q&A panel. To have your questions addressed during this time, please post them in the Q&A panel function in your Zoom window as we move through the talks. It would be great if you could also include your affiliation as well as the speaker your question is directed towards with your question. I see. Oh, okay. Eugene, no echo. Okay. Okay. If there is so let me go back over that again. To have your questions addressed during the Q&A panel, please post them to the Q&A panel function in your Zoom window as we move through the talks. It would be great if you could include your affiliation as well as the speaker the question is directed toward with your question. If there is a question already posted in the Q&A panel and you have the same question, use the Zoom upvote function on the similar question to help ensure we get answers to the most popular questions during the panel. Please note the Q&A panel function is separate from the chat function in Zoom. Again, questions for the presenter should be posted in the Q&A panel. If you're having technical difficulties during the symposium, please let us know via the Zoom's chat function. And we will be happy to assist you. Following the Q&A, we will break for lunch and resume at 12:45 for session 2. All symposium presentations are being recorded and will be posted to the Algal Bloom Action Team website at the conclusion of the event. A special thanks to our partner level event sponsors, FlowCam, Limnotech, and Eutrophics. Your support allows the Algal Bloom Action Team to provide future harmful algal bloom events and educational activities. Again, this morning's presentations focus on harmful algal bloom monitoring and ecology, and I will now turn it over to Eugene to introduce our speakers this morning.
Thank you, Amy. Again, my name is Eugene Bragg, and I work for Ohio State University Extension. I'm Program Director for Aquatic Ecosystems. By training, I'm a fisheries biologist, but in practice, I'm a limnological jack of all trades. And before I introduce our speakers, I do want to remind you to please use the Q&A function to ask your questions. As you ask a question over the course of the day, it would be helpful if you also type a specific speaker to whom the question is directed if you have a specific speaker in mind for your question. We're doing it a little bit differently this year in that we will have a closing question and answer panel with each session over the course of the next 2 days. Use that upvote feature to move up the priority for questions that are already posted and that you feel are important, just so we're sure that we get to them all. And you will not be able to unmute yourself as a participant today, so make sure you make use of that Q&A function. So first, I'd like to introduce Matt Hudson. Do you mind advancing the slide, Amy? So I'm not going to read the full bio. It's there for your consumption because I'd much rather get to Matt's talk. Matt is an associate director and water scientist with Mary Griggs Burke Center for Freshwater Innovation at Northland College. And today he is going to speak on the evolving story of cyanobacteria blooms along Lake Superior's south shore. So Matt, welcome. Thank you so much for being with us today.
All right. I'm going to share my screen here. How's that look, Eugene?
Seeing the presenters mode, so we're seeing what you're about to see as well.
Okay, so you have my title slide?
I do, but it's in, again, presenters mode, so we're seeing a preview as well as any notes you might have.
Oh, okay. Let me try this again. All right, share. How about now?
That looks like just your title slide. Looks good.
All right. Not too bad on the technical difficulties, but thanks everyone, Anne and Amy, all the organizers for this conference, for the opportunity to speak here today. Again, I'm Matt Hudson, Associate Director and Water Scientist with the Burke Center, which is at Northland College in Ashland, Wisconsin. So we're located way up on Lake Superior, about as far north in Wisconsin as you can go. And our college, our little college up here is primarily a teaching institution, but the Burke Center is an applied learning opportunity for our undergraduate students. We provide opportunities for folks in freshwater science and communications. So I want to acknowledge my co-authors on the talk here, Brenda Marasca-LaFrancois from the National Park Service, Kate Reinl from the Center for Limnology at UW-Madison, Liz LaPlante with the EPA in Chicago, and also Bob Sterner with the University of Minnesota Duluth Large Lakes Observatory. Bob provided a lot of content that we'll be seeing in the presentation, and really Brenda, Kate, and Bob are the pioneers in a lot of what's been going on in terms of understanding the evolving story of cyanobacteria blooms in Lake Superior. And so a lot of the credit really needs to go to them and to others. But I'm happy to carry the torch today here and tell you all a little bit about what we're learning about this surprising phenomenon of blue-green algae blooms in the largest of the Great Lakes. So let's start with that. Lake Superior, for those, I know we have an audience that's from a fairly wide geographic range here, so many of you may not be familiar with the Great Lakes and especially Lake Superior. Here's a satellite image of the lake. Our Duluth is over here way in the western end, and Ashland and Northland College are just to the east in Chequamegon Bay right here. And so Lake Superior is an immense body of water by any measure. It's the largest surface water freshwater lake in the world by surface area and third largest by volume. It contains more water than all of the other Great Lakes combined. And if you spread that water out over North and South America, there'd be about a foot deep of water across that entire land area. So it's just an immense amount of water that's in this lake, and its size cannot be understated. So when we think about Lake Superior in the context of the rest of the Great Lakes. Here's the Great Lakes Basin on this figure here from Dale Robertson. And the Great Lakes have this wide range of trophic states from Lake Superior at the headwaters area, which is largely a forested, low urban and agricultural development, and thus low amounts of phosphorus in the water column, typically in Lake Superior, whereas areas like Green Bay, Saginaw Bay, Maumee Bay, these are some of the areas of the Great Lakes that have typically struggled with issues related to HABs. And so the land cover is quite a bit different when you get down into areas more in the Lake Erie, Southern Lake Huron, Lake Ontario basins. So we have this wide range of trophic states that leads to a wide range of water quality conditions in the Great Lakes, from very turbid, of course here we've got a cyanobacterial bloom in Lake Erie, which are very common, over to Lake Superior, which has legendary clear water. And so quite a bit of difference there, related again to a lot of what we see in terms of land cover differences between these different parts of the Great Lakes Basin. A little bit more for you about Lake Superior. It's just an amazing resource that people love to come and visit. It's generally regarded as very pristine and picturesque, and we have some photos here. This from Canada, Nipigon Bay, large towering cliffs like these in pictured rocks, epic sort of Caribbean style sand beaches in the Apostle Islands. Although this time of year, maybe being in the Caribbean would be a better place to be if you don't like snow. And we just got about 20 inches last night, by the way. And so here, this photo here is of the Kakagan Bad River Slough. So there's a lot of these highly intact coastal wetland resources in Lake Superior. So by many measures, the most pristine of the Great Lakes. However, this satellite image shows the western part of Lake Superior, again, Duluth over in the way western tip there, and Ashland here in Schwamingen Bay. So this part of Lake Superior has, because of its surficial geology and its land cover, land use history, so this area was clear cut about 100 years ago, and so that interaction of surficial geology and land cover has led to a lot of erosion and sedimentation issues in this part of the lake. So these kind of sediment plumes like this often do appear during high runoff events in this part of the lake. This image is from 2018, and I'm going to be talking a little bit about how some of those large storm events seem to be increasing in this area. But while sediment plumes are not unexpected in this part of the lake, we certainly didn't anticipate that Lake Superior nearshore would ever look like this. So this photo you might more typically associate with Lake Erie, but this is a photo from Lake Superior from 2018 taken by my co-author Brenda. And this issue has gotten a lot of regional and national attention in recent years too. The New York Times did a feature a few years ago. The Chicago Tribune had a front page story just this past fall, and there's been a range of regional print and TV reports about this issue. So it's really captured the public's attention with respect to, wow, this is something we didn't expect to see happen in the largest and cleanest of the Great Lakes. So this graphic here gives you an idea of the bloom reports and sort of what's been going on to date. And so there is these open circles here are some historical anecdotal observations, but they weren't ever confirmed under a microscope or anything like that. So this area on the way western end of the lake in the 60s, there was mine tailings being dumped into the lake, and any blooms that were observed during this time were likely due to iron enrichment from those mine tailings. There hasn't been any recent blooms in this area. And then there were a couple of reports from kayakers in the Apostle Islands of possible blooms in the 80s. But again, nothing was actually confirmed until 2012. And you can see that again in this western part of the lake, where we see a lot of the sediment plumes, is where the vast majority of the bloom reports have been occurring since 2012. And there's been at least a small bloom reported in just about every year since 2012. And in the last couple of years, there's been a couple of reports here along the Canadian North Shore of small blooms as well. So this seems to be a recent phenomenon that's been occurring within the last 10 years. And it seems to be largely focused in this region in the western part of the lake along the Wisconsin shoreline. Here's some photos of what these blooms tend to look like. And when you see Sea Caves, Corny Beach, Myers Beach, all these places are again located in that area I just showed you along the south shore of the lake. And the blooms tend to be, again, range in size from very small, localized blooms, surface scum, floating algae within the water. They have presented themselves in different ways. This picture from Corny Beach in 2018 was the most extensive bloom, where large portions of the water column were affected. But in many ways, we see these blooms as somewhat typical as you might see in other places. The 2018 bloom was the most extensive bloom. This basically went from the star over here near Duluth Superior along the near shore of the lake all the way up, and it was most prevalent up here towards the tip of what this is the Bayfield Peninsula, to the extent where we could detect the bloom from space. And so there was one report of a small bloom over here to the this is Madeline Island over here, but by and large most of the activity was along this southern shore. I guess it'd be the northern coast of the Bayfield Peninsula in Wisconsin. So this bloom is what really caught the attention of many folks just because of its size and the recurrence of these blooms. Wasn't just a one-off thing anymore. These things have become more common. And so the dominant species that that has been a part of all these blooms is Dolichospermum lemmermanii. You can see some pictures of it here from Brenda on the right. It's a filamentous colony-forming cyanobacterium, and it has caused blooms in other oligotrophic systems. There's some research from the Italian Alps that suggests blooms of this species may be increasing in some of alpine lakes in that part of the world. So we frequently see the heterocysts and akinetes being present in these cells from Lake Superior. So the nitrogen fixing heterocysts and the resting cells, the akinetes. And lemmermanii is also a potential toxin producer. So let's talk about that. So the blooms to date do not appear to be toxic. Toxins are generally below detection in samples that have been collected. However, not a lot of samples have been collected and not all potential cyanotoxins have been analyzed. And some potentially toxic substances such as anobenopeptins have been detected in samples, although I think the toxicity on these is still a bit up in the air. So elevated microcystin was detected in a small bloom that occurred in the Duluth Superior Harbor. So this is a harbor area that's isolated from the main part of the lake, but this was the first time that this toxin was detected in any of the connecting waters to Lake Superior. But I guess the bottom line here is that the potential for toxin production from these blooms is something that needs further exploration. This is something that Todd Miller from the University of Wisconsin-Milwaukee has been actively involved with and is continuing to work on. Okay, so what do we know about why these blooms are occurring? So far, we're pretty early in the research, but mainly the evidence is mainly pointing to climate-related drivers. So in Lake Superior, land use change is not likely a driver because there really is very little agriculture and urban land cover. About 70% of the watershed is in forested land cover, and that really hasn't changed over time. So this is very different than other places in the Great Lakes where we see issues with HABs. So land cover, land use, not likely the culprit here. Current evidence and projections are for a warmer, stormier future, however, and that's not terribly uncommon for other areas of the country and the world. But one thing about Lake Superior is that this recent study in 2015 identified it as one of the fastest warming lakes, large lakes on the planet. And so Lake Superior is warming very rapidly. And part of what that's due to is, if you look at this figure on the right here from John Lenters, The onset of thermal stratification, particularly in years like 1998, 2012, which are highlighted here as years that were particularly warm. And so the water column was able to stratify earlier in the early part of June. And once that happens, then that surface water can really warm up more rapidly as compared to colder years where we see the stratification didn't set up until into August. So when you got, more opportunity for warmer water, especially water that gets up into the 20-degree range where we start to see the opportunity and the potential for species like Dolichospermum lemmermanii to bloom. This figure, again, another one from Bob Sterner. The biggest Lake Superior blooms have occurred in the years with the warm water temperatures. So what this figure is showing is the accumulated degree days over the course of several years here. The blooms are indicated, years with blooms are indicated, the large ones in red and the smaller ones in blue. So by and large, bloom years tend to also be warmer years, warmer water years. So climate change appears to be stacking the deck for more of these warm years moving forward. So temperature is only part of the climate story. Storms are also part of the factor here. And I showed you this aerial image of the sediment plumes in the western part of Lake Superior. This was following a historic rain event in June of 2018 that led to a lot of infrastructure failures and a lot of erosion and sedimentation that ended up manifesting itself in these large sediment plumes that lingered in the lake for weeks following this rain event. So 2012 and 2018 were the years with the largest blooms in Lake Superior. There was those blooms were preceded 25 days in 2012 and 53 in 2018 by these very large storm events. These figures give you an indication of the recurrence interval, the areas where, for instance, in Duluth here, this is some of the depth of precipitation that was received here was about a 500 recurrence interval. Other areas in Northwest Wisconsin received up to 1,000-year recurrence interval. There was another large event in 2016 as well that was a little bit further over to the east that also was near 1,000-year recurrence interval precipitation event. So 3 of these really large storms in just a few years is really giving us an indication, okay, this is what's being predicted for a changing climate, and maybe we're starting to see the manifestation of that here with these large events. So why that's important is during high flow conditions, during these large events, this is where most of the phosphorus and nitrogen, not only are they elevated, but the volume of material that's delivered to the lake is much greater during storm events than at base flow. So on the left, we have nitrogen loading. This is in the Brule River, which is one of the tributaries along the south shore of Lake Superior. Phosphorus increasing with discharge. And when we look at this, we see that, a large event that's delivering, potentially 40 times greater than, or even more than what we typically see in terms of phosphorus loading at base flow. So these large events just deliver these episodic pulses of nutrients, sediment to the lake. And they also may be delivering and supplying some of the necessary cyanobacteria propagules to the lake in order to seed some of the blooms that we're seeing. So this is research here from Kate Reinl, one of my co-authors. Her PhD research was looking at the fluvial seeding of cyanobacteria blooms in Lake Superior, and she did a series of laboratory growth experiments from river areas, harbor areas, and within Lake Superior itself. And the only place where cyanobacterial growth occurred were from these harbor and river samples. So that's an indication that the propagules, or the cells capable of growing in the laboratory anyway, were only present in these upland areas and not from Lake Superior itself. So we've also learned that blooms tend to occur in some places, but not in others. And this is an interesting question: is what is driving that? And so part of Kate's research here, this figure on the upper left, she found that rivers with high conductivity and low temperatures had the greatest blue-green algae growth in the laboratory cultures that she ran. So the hypothesis here is that Either these rivers are transporting the cells from other upland areas, or they exist within the rivers themselves. But when they get out into the lake where it's actually warmer during the peak of summer temperatures, this provides an environment where these cells are potentially able to bloom and thrive. And this figure on the right is, again, you go to the other side of the Bayfield Peninsula. This is Chequamegon Bay. This is where we're located. So the Burke Center has done some research to replicate Kate's work. We haven't seen cyanobacteria blooms in Chequamegon Bay, but this is one of the areas of the lake you would expect to see these kind of blooms happen first. This is a warm, shallow, fairly isolated embayment from the large part of the lake. There is a lot, in terms of Lake Superior anyway, a fair amount of agricultural land use within the watershed. Yet when we run the same culture experiments, we don't get much in terms of blue-green growth from any of these areas around Chequamegon Bay. With the exception of over here to the right, this is the Bad River, and the one bloom that has been reported over in this region off between these two islands here was in 2018. The Bad River was a place where we did see some growth. So anyway, there's a story evolving here about, okay, what exactly is it that leads to blooms forming in some places and not in others? We did do a bunch of, collected a whole bunch of samples across the entire near shore of Lake Superior this summer, and we're looking at microbial DNA sequencing to try and get a better idea of how the microbial community varies spatially and temporally across this region to see if that gives us more clues into why we see them in some areas and not others. All right, now I'm going to move from the science of what we know to the response from the research and management community to these blooms. And so the response has been very extensive and coordinated. Again, the National Park Service, Brenda and Kate and Bob, Sandy Bravold from University of Minnesota Duluth Large Lakes Observatory really led the way here. They established the first sites along the near shore of the lake, as well as sampling for nutrients, biomass, algal composition, toxin. They brought in Todd Miller, and they've deployed some continuous sensors for continuous monitoring, the lab culture experiment work, and now they have some sediment cores that were just taken this past summer to look into the sediment record to see if there's evidence of past blooms or if this really is a new phenomenon within the lake. So again, the Park Service and UMD were really the leaders here, and many other agencies and universities have joined forces. We got involved in 2019, replicating the Kate Reynolds experiments because we hadn't seen blooms in Chequamegon Bay and we're wondering why. The Wisconsin Department of Natural Resources, US Geological Survey, and the National Estuarine Research Reserve in Lake Superior all did an extensive nearshore sampling in 2019. And around the same time, in 2018, following that really massive bloom that occurred, the Lake Superior Partnership Working Group. So this is all of the Great Lakes have a LAMP, or a Lake-Wide Area Management Plan, basically a group of the federal, tribal, state agencies that work together across the Canadian-US border to manage each of the Great Lakes. And so it was recognized early on that this LAMP working group needed to pay attention to this algal bloom issue, and a special work group was formed. So the state agency, the people involved in the LAMP could communicate with the scientific community. And also a bloom rapid response communication plan was developed through this group, including a phone tree, and sampling kits were sent out for species ID and toxins. So we needed to be able to develop a way to get people out on the water if a bloom was spotted, and get as much sampling and information as we could. Many of these blooms have been very short-lived in nature, so there was definitely a need for a rapid response plan, and that's what we've been doing through this group. And the rapid response was initiated a couple of times last summer and led to some good sampling. Okay, so regional policymakers have also taken notice. The International Joint Commission, or IJC, which is a body that makes recommendations on issues related to transboundary water management between the United States and Canada. So the IJC provides recommendations directly to the governments of the US and Canada on transboundary water issues. And the Great Lakes, of course, are an area of particular importance because of their immense size. And so the IJC is required to do some listening sessions across the Great Lakes, and they came to Northland College in 2019, as well as UMD and Thunder Bay. And they got an earful from people over here that this algal bloom issue was really concerning, and we need to learn more about it. We published a white paper on the proceedings from that event, and the IJC responded with their triennial assessment of progress report to the governments of US and Canada that the government they recommending the elimination of algal blooms on Lake Superior. So this issue was elevated from a policy standpoint by the IJC's report. And so the elevation of the issue has also led to an infusion of additional resources to study it. The Cooperative Science and Monitoring Initiative is another, I guess it's a part of the work of the LAMPS and the Great Lakes Restoration Initiative. But there's a 5-year process that occurs in each of the Great Lakes where there's a field year. So each of the lakes gets an infusion of funds for an intensive year of sampling to study particular questions of interest in each of the Great Lakes. And then there's a analysis, reporting, priority setting, and planning. So it's a 5-year cycle for the CSMI. And 2021 happened to be Lake Superior's year of intensive monitoring. And so there was a large group of agency and university and other folks that have come together to study this issue and collaborate on figuring out what are the drivers of these blooms. This just gives you a sense of the different kinds of work that's going on and some of the players that are involved. The summer of 2021 was a very, very busy year. A lot of boats on the water, a lot going on mainly in the Wisconsin, this area where the vast majority of the blooms have been occurring, looking for tributary signals, looking within the nearshore itself. The microbial DNA sequencing to look at the community from that perspective. Some really detailed landscape management modeling being done. And then the sediment coring. And so now we're moving into the stage. Oh, sorry, before I get to that, this kind of gives you maybe a sense of the scale of the different work going on. All these dots on the map represent a place where somebody was doing something related to the CSMI project. And so this ArcGIS Online tool was used as a way for us to see who was doing what and where, so it could help promote coordination and collaboration. Because when we get into the midst of the field season, to be able to have something like this, to know who's doing what, it can be a good way to respond and adapt and evolve sampling programs as needed, depending on what's going on the ground. Okay, so last month we held a sort of a report out from what happened in the 2021 Coordinated Science and Monitoring sampling. Kind of informing each other where we're at and starting the conversations about collaborations during the data analysis phase. And so the hope here is that enough manuscripts will be developed that we can pitch a special issue for the Journal of Great Lakes Research to help capture this research that's being done this summer, not just on the algal bloom issue in Lake Superior, but some of the other year of intensive monitoring projects as well. Okay, so let's wrap everything up here. With some of the take-home messages. I've thrown a lot at you today, but number one, Lake Superior, for a long time was thought to be sort of immune from human influence just because of its vast and massive size, and that's being proven wrong again and again. And certainly this algal bloom issue is another example that Lake Superior is vulnerable to detrimental change. The blooms were unexpected, and they really highlight gaps in our monitoring and knowledge of the Lake Superior ecosystem. We just didn't see these coming, and so we're kind of behind the 8 ball a little bit in terms of understanding what's going on here. And so hopefully the issue is going to improve our ability to respond to these blooms moving forward. Evidence is mounting that climate change is really one of, if not the main driver here of these blooms. And so the lake is warming very rapidly. There seems to be an increase in these very large storm events that deliver episodic pulses of nutrients, and perhaps the upland propagules, the upland systems may seed these blooms with large storm events. We may see more of that material being delivered to the lake as well. So Lake Superior may provide an opportunity to look at more climate-driven influences on large lakes, especially related to HABs and blue-green algae blooms, because the amount of urban agricultural land cover that is just so low in Lake Superior as compared to places like Lake Erie, Green Bay. So we may be looking at more of a pure climate signal here. And so, as we're learning more about these blooms, and we're also going to be learning about what can we maybe do about them. Are there opportunities to implement best management practices to help reduce the likelihood of these blooms occurring? Chances are this is probably something that we're going to be seeing more and more of in the future if climate predictions continue to play out. But as we learn more about this issue and where these blooms are coming from, there's hopefully some things we can do about limiting their influence. Because Lake Superior, of course, part of its allure and its draw is its legendary clean, clear waters, and so there's definitely concern that if Lake Superior becomes heavily burdened by blue-green algae blooms, that's going to hurt a lot of the economy in this region, which is in many ways dependent on tourism in many areas. Okay, so what will the lake tell us next? We'll continue to figure that out as we go through all the data we've been collecting and gather more in the coming years. But I really need to list all these folks here. Special thanks to so many people that have been involved in the blue-green algae research and the funding through GLRI and the Wisconsin Coastal Management Program, BRECO Fund, and many more. I'm sure I've missed some folks that have been involved in this issue, but it's been great to be a small part of working with so many great people and trying to figure out, what's going on with this particular concern, HABs in Lake Superior. So that's all I have, Jean. Appreciate the opportunity. I'll end my slideshow here.
And don't forget to stop sharing your screen as well.
Yes.
Excellent presentation, Matt. And I have so many questions myself, but in deference to our attendees, I will, at the panelist session at the end of today's session, this morning's session, I will defer to our attendees. Thank you so much, Matt. That was an excellent presentation. I think very informative. I'm going to offer a quick reminder before we move to our next speaker that if you do have questions for those late attendees, if you do have questions for any of our speakers, please use the Q&A feature that you're going to find in the Zoom toolbar. If it's specific for a particular speaker, please name that speaker and use that upvote function. It looks like we're getting a lot of questions. I doubt we'll be able to address all of those questions. So if you see a question that's already posted, that is something you're particularly interested in having answered, please upvote that question. That moves it up in the priority list, and we will address them in order of priority using that upvote feature. So in the interest of moving on to our next excellent presentation, I'd like to introduce Heather Krempa. Heather's a hydrologist for the USGS in Lee's Summit, Missouri, an aquatic biologist with a passion, as she says. But once again, I'm much more interested in getting to her presentation than in reading her biography. So her biography is on the screen for your education. Heather is going to discuss her shop's program for the Illinois River Basin HAB initiative. So Heather, welcome. If you're ready, feel free to share your screen and begin.
Okay. No. Can you see my screen?
I can, it looks good.
Okay. And you're adjusting the title slide?
Yep, just the title slide.
Okay, great. Thanks for that introduction. As Eugene said, my name is Heather Krempa. I am a hydrologist for the U.S. Geological Survey, Central Midwest Water Science Center. That includes Iowa, Illinois, and Missouri. I'm located in Kansas City, Missouri. However, today I'll be talking with you about several USGS HABs programs, but most specifically the Illinois River Basin, Cyanobacterial Harmful Algal Bloom Initiative. So I'm not going to spend a whole lot of time on a background for harmful algal blooms, right? That's the reason we're all here. However, I do want to point out that although several types of algae can cause harmful algal blooms, I will be focusing specifically on cyanobacterial harmful algal blooms. And I will refer to them as cyanoHABs frequently throughout the presentation as well. So cyanobacteria comprise cyanoHABs. They are true bacteria, however, they have photosynthetic capabilities, so they're generally considered part of the algae community. And they're also known as blue-green algae. Although blue-green algae are microscopic, they can form blue-green mats or swirls in the water that many people refer to as looking like spilt green paint. Seen very clearly here in the photo, and they are motile, so they can move throughout the water column and are not always aggregated at the surface.
Heather, I'm going to warn you before you move too quickly that your audio is breaking up a little bit. So if there's anything open that might be gobbling bandwidth, you might want to do a little bit of trimming.
You guys are not seeing my slide anymore, are you?
No, but now your presentation's disappeared.
Is that back now?
It's back.
Okay. I will try and slow. I am not in the location I expected to be to give this presentation, so hopefully we can make it through. But
I still appreciate it, and it's minimal. So carry on without concern. The audio breakup is minimal, but it is there.
Okay, great. So moving on to what makes a algal bloom detrimental to economics, especially in communities that are close to the water. However, they are also harmful directly to humans because they are capable of producing cyanotoxins. These cyanotoxins are directly harmful to humans, pets, and wildlife. And they are also known to be the cause of pet deaths, including dogs that have drank contaminated water. And humans can be exposed to these cyanotoxins through drinking water, ingestion of contaminated food, which includes fish, and by inhalation or by skin contact. And exposure to cyanotoxins can cause many symptoms, including abdominal pain, headaches, nausea, diarrhea, skin rashes, respiratory issues, and seizures. And there's also evidence that there can be some larger issues such as tissue disruption to the liver, kidneys, and spleen, and possibly some carcinogenic effects with long-term or repeated exposure. This is kind of what makes me nervous as a HABs researcher, because you start to consider how much additional exposure you're giving yourself by being out here sampling these HABs, knowing that they can be potentially toxic. You take the best precautions that you can, but sometimes you find yourself eating a peanut butter and jelly sandwich on the Illinois River using a clean glove to reduce that contamination. HABs are harmful to many other aspects of human life, including the economy. Cyanobacteria can also produce taste and odor compounds. These can build up in fish tissue and can be tasted in drinking water. So there's additional drinking water treatment costs. And lastly, the overall aquatic ecology can suffer from cyanoHABs, especially because as the cyanobacteria biomass die off, they decompose, which consumes oxygen, and reducing the dissolved oxygen concentrations can lead to fish kills and the death of other aquatic organisms, and altering natural food webs and reducing overall biodiversity. So harmful algal blooms are harmful in lots of ways and affect human life in many ways. And with most science topics, as you start to consider them, you realize that there are always some things that we know, more things that we don't know, and then there are always those things that we don't even know that we don't know. And as you continue to study a subject, you expect that you learn a little more about what we know and you take some things away from that what we don't know column. However, that doesn't seem to be the case with cyanoHABs. The more we study cyanoHABs, it often seems like the only consistent conclusion is that they're complex. CyanoHABs are generally associated with excess nutrients, warmer water temperatures, high light availability, often stagnant waters. However, this is not always the case. The conditions that trigger the occurrence, duration of, and toxicity of cyanoHABs seemingly can be different between lakes and rivers, between different water bodies, between different years, even between different months. We do know that there are several genera that can produce cyanotoxins and taste and odor compounds, but these are strain dependent and not necessarily genus dependent, and therefore the toxin producing genera can be within the cyano bloom, but not have the strain that is capable of producing toxins. Also, we know that the strain that is capable of producing compounds may be present, but they may not be producing those toxins or the taste and odor compounds. Therefore, the presence of the correct strain doesn't always indicate that a bloom is toxic, simply that it has the potential to be toxic. On top of that, there are multiple cyanotoxins and multiple taste and odor compounds that can be produced and that can coexist. This means that while studying cyanoHABs, different aspects of the cyanobacterial community need to be sampled to understand if cyanotoxins and taste and odor compounds are capable of being produced, which compounds are capable of being produced, and if and what those compounds are being produced. Also, blooms can form and dissipate or move quickly. Therefore, the cyanoHAB occurrence and the toxin potential can change very rapidly. So as I said, it seems like as we continue to study cyanoHABs, the list of things that we don't know is increasing more rapidly than the things that we do know. Although cyanoHABs are often associated with excessive nutrients and warmer water temperatures, they're becoming more frequent in oligotrophic lakes and under ice, or are at least being noticed more frequently in these areas. The conditions that cause the cyanoHAB to occur and those that control the toxicity, magnitude, and duration are still unclear. These conditions are important to understand the best predictors of HABs occurrences and to develop predictive tools or advanced warning systems. Currently, it can take several days to get lab results back to warn the public if a toxin is present in an area. There are a lot of new technologies. Being explored to better understand what conditions are driving HABs occurrences and toxin production. These include many real-time sensors and also the use of spectral imaging from multispectral cameras, satellite imagery, and also eDNA capabilities are starting to be explored. Other questions that are out there are what happens to the cyanoHAB when it is no longer visible? Is it still toxic? Are the cyanobacteria still present but distributed through the water column? Are they transported downstream? Do the toxins persist or do they get transported? And finally, as I mentioned earlier, that there's still a lot unknown about long-term exposure. As cyanoHABs continue to develop in areas of recreation and make their way into our drinking water and our food sources, there may be more understood about the long-term effects and accumulation in tissues. And then there's always that inevitable, what we don't even know that we don't know. These ideas, however, will continue to be developed, and I appreciate symposiums such as this that bring science together and scientific thought, and through continuing the sharing of information and ideas through conferences such as this, and fieldworks, publications, and other aspects, we can start to kind of figure out what those aspects are that we don't even know that we don't know. cyanoHABs are continuing to be of increasing concern. They are seemingly to be more frequent, or they're at least being noticed more, and public awareness is being increased. Because of this, the USGS has developed several programs that are dedicated to, or at least include an aspect for studying cyanoHABs. I'll briefly discuss several of these USGS programs, and then I will end in more detail with the Illinois River Basin NGWAS Cyano-HAB initiative. Starting with the National Water Quality Program. This program began in 2019 when Congress provided additional cooperative matching funds to assess HABs. These cooperative matching funds are used for projects that are done in cooperation with state, regional, tribal, or other local partners. The National Water Quality Program is funding 24 projects in 15 geographic regions, which are those shown in blue on the map here. The goals of these projects were to focus on early warnings of HABs by advancing real-time monitoring, remote sensing, and the use of molecular techniques for identifying and predicting the occurrence of HABs and cyanotoxins. Also, projects that included the use of traditional and emerging technologies were given priority. The second USGS program that I'll discuss is the HABs Proxies project. This project includes a team of approximately 15 scientists from across the USGS and the nation that have been tasked with developing proxies for harmful algal blooms. How a harmful algal bloom is defined in terms of what is considered harmful, which algal taxa are of interest, and what counts as a bloom affects what data are collected and how those data are used in analyses and modeling. An early goal of the HABs Proxies project was to codify language around these various aspects of HAB definitions by compiling and evaluating published metrics and thresholds from scientific and from gray literature. The project will also use in situ water quality information from USGS sites. These data will then be used to develop what are known as proxies for HAB occurrences in rivers and to better understand temporal dynamics of these events. Additionally, these proxies will be linked to environmental conditions to identify river reaches that are vulnerable to HABs. This is a 4-year project that is currently in year 2. It focuses on rivers, not streams, and HABs in terms of excessive algae biomass, not defined by being toxic. The team is beginning in the Illinois River, but plans to expand to the Ohio and then the upper Mississippi River basins, with hopes to being able to expand this nationally. During the first year of the HABs Proxies project, they focused on evaluating approaches for developing proxies, and an internal feasibility assessment was developed, detailing a work plan and identifying project goals. This is currently year 2 of the project, and the team has retrieved, processed, and summarized USGS and water quality portal data for the Illinois River Basin. Data pipelines that streamline these processes have also been developed that will be used as the project expands its geographic reach. Currently, the team is working on 2 of its first projects. An initial product will compile and evaluate HAB definitions, related metrics and thresholds. They'll describe how proxies conceptual framework can help clarify issues with HABs definitions and modelings, and apply these concepts to the Illinois River Basin. The second project will focus on building machine learning models at 6 data-rich sites in the Illinois River Basin, and then extend those models to sites with similar data in Ohio and upper Mississippi River basins. The goal of this product is to identify common metrics that can be used across a variety of locations, hopefully overcoming the site-specific dependencies of many surrogate-type models. For example, if a proxies model is identified that uses water temperature and dissolved oxygen, this model could be applied at locations that have those same data, including the sites that are shown on this map here. Jenny Murphy is the team lead for this project, and she can be reached at the email that is located on this slide if you have further interest or questions. And the final USGS project that I'll be discussing in more detail is the Next Generation Water Observing Systems, or NGWAS. The goal of NGWAS is similar to that of the National Water Quality Program. However, these projects focus specifically on a defined water basin, whereas the National Water Quality Program funded several projects in 15 different geographic regions. NGWAS was developed to focus on advancing water science with emerging breakthroughs in technical and computational capabilities to provide real-time water quantity and quality data in more affordable and rapid ways than previously possible, and in more locations. And this goal of providing real-time water quality data and quantity data in more affordable and rapid ways and in more locations will be accomplished using state-of-the-art measurements, arrays of sensors at selected locations, increasing spatial and temporal resolution. New technology will be implemented and tested to improve future operational efficiency, and there is also a focus on modernizing data storage and data delivery. NGWAS started in the Delaware River Basin during 2018 with a focus on new stream gauges to fill critical gaps and enhance the water monitoring network. Since it was initiated, there has been intense monitoring, including remote sensing from drones, advanced camera use, and testing of non-contact sensors. The Upper Colorado River Basin was the next NGWAS basin selected, and this basin was selected to focus on improving regional water prediction in a snowmelt dominated system. The final NGWAS basin that has been added is the Illinois River Basin, which was added in 2018 with the goal of filling monitoring gaps in data needs and to advance the understanding of nutrient delivery and harmful algal bloom prediction. So now I'll focus specifically on the Illinois River Basin NGWAS project. The Illinois River Basin contains about 44% of Illinois. It includes parts of includes parts of Wisconsin and Indiana, and it is an important transport mechanism between the Great Lakes and the Mississippi River. The land use includes both urban and agricultural. This includes Chicago as a major urban city in the upper parts of the basin and corn and soybean agriculture in the lower parts. Again, Illinois River Basin was selected specifically for the next NGWAS basin because of its potential to fill many data gaps, including information about harmful algal blooms. There is historical evidence of cyanoHABs in the Illinois River. Looking at Google Earth images from 2015, there appears to be a cyanobacterial bloom occurring. This is Google Earth images of the same location where a cyanoHAB was confirmed in 2020 by the Illinois EPA. The Illinois EPA received information of a potential cyanoHAB in this area. The water was tested for microcystin and a concentration of 138 micrograms per liter was determined, which is well above the recommended recreational level of 8 micrograms per liter. The Illinois EPA did put out a press release informing the public of the potentially toxic water. And there are also many unconfirmed statements of reoccurring cyanoHABs within the Illinois River Basin from other state agencies. Knowing that cyanoHABs are likely a reoccurring problem within the Illinois River Basin, objectives were developed for an NGWAS Illinois River Basin cyanoHAB initiative. While developing these objectives, the things that we don't know about cyanoHABs were considered. Since the Illinois River Basin is generally associated with high nutrient concentrations, and at this point, there's little evidence of cyanoHABs under ice, this first bullet was not a focus. However, the evidence of recurring cyanoHABs in known locations within the Illinois River Basin, it was likely that this basin would provide patients to study conditions that trigger cyanoHAB occurrences and those that produce the cyanotoxins and the taste and odor compounds. Taking this into consideration, the objective to identify physical, chemical, biological, and hydrological and meteorological conditions prior to, during, and after cyanoHABs to improve the understanding of the conditions driving cyanoHAB occurrences, magnitude, duration, and cyanotoxin and taste and odor production was developed for the NGWAS Illinois River cyanoHAB project. This will be accomplished using several continuous real-time water quality sensors, including those that measure temperature, dissolved oxygen, specific conductance, pH, chlorophyll, phycocyanin, nitrate, and phosphorus, and by collecting samples for algae community, chlorophyll, nutrients, sediment, cyanotoxins, and taste and odor compounds. The Illinois River Basin may also be an ideal location to test and develop new technologies and tools that could be used to advance warning prediction potential. Therefore, 2 additional objectives were developed, including to improve remote sensing of total chlorophyll A, and phycocyanin concentrations by installing and evaluating various sensors and by using multispectral cameras at USGS supergauge locations and at mobile locations to evaluate multispectral imaging for monitoring, identifying, and predicting cyanoHABs. Also, eDNA capabilities will be explored. Since NGWAS is focused on the Illinois River Basin, effects including bioaccumulation were not focused on at this time. However, the next focus points of the fate and transport of cyanotoxins in tastes and odors and toxicity when cyanoHABs are not visible were considered. This led to the objective of collecting samples before, during, and after occurrences, which will include sampling at regular intervals to ensure that samples are collected before cyanoHAB occurrences, which will give us a better picture of the physical, chemical, and biological conditions that lead up to a cyanoHAB occurrence and what happens after the cyanoHAB is no longer visible. Samples will also be collected upstream and downstream of the visible cyanoHAB to better understand the spatial distribution. Samples that would need to be collected to accomplish these objectives were considered, and a long list of samples was developed, including nutrients and sediment, phytoplankton and periphyton community structure, chlorophyll A, phyophytin A, and ash-free dry mass, cyanotoxins, including microcystins, cylinders, spermopsins, anatoxins, and sex toxins, and many taste and odor compounds. Also, we'll be collecting samples to compare with hyperspectral imaging, including cameras and satellite imagery. Continuing to consider these objectives, a sample plan of when and where to collect samples was developed and includes sampling before, during, and after a cyanoHAB event is visible at locations upstream and downstream of the visible cyanoHAB. CyanoHABs will include surface grab samples at single locations, compositive surface grab samples across a cross-section of the river, and depth integrated samples to compare the differences in conditions at the surface and throughout the water column. And as you can see from this picture of a subset of those samples, many samples are necessary to describe the spatial and temporal variability. These samples will also be collected at super gauges where several types of sensors will be deployed throughout the year. Sensors that will be deployed within the water column include temperature, dissolved oxygen, specific conductance, pH, chlorophyll, phycocyanin, nitrate, and phosphate sensors. Also, an algae tracker will be installed, which deploys sensors at the surface of the water as opposed to at an established depth. These sensors include green algae, cyanobacteria, turbidity, temperature, and sunlight. Since historical blooms were identified through Google Images and IEPA samples close to the established USGS gauge at Starved Rock, this was the first super gauge established through NGWAS. Other gauges that will be important include the gauges upstream and downstream of Starved Rock, which include Seneca and Henry. Also, a mobile unit will be developed with similar sensors to be used at non-established sites of interest where samples will also be collected. And like most things with field work, just as our sample plan was being developed, we got an opportunity to test how prepared we were. On June 9th, the U.S. Army Corps of Engineers reported a potential cyanoHAB near Starved Rock to the IEPA, who reached out to the USGS, allowing for collaboration among these agencies because cyanoHABs don't wait for you to be ready. The day following the report of the potential bloom, the IEPA was able to collect samples for cyanotoxins and made a visual confirmation of the bloom. The day following that, microcystis species were identified in the samples, which is a common cyanotoxin producer. During June 15th through 17th, the USGS implemented their, at this time, preliminary sample plan, and we collected samples around the Starved Rock pool and where the bloom was not visible, upstream and downstream. On June 17th, 8 days after the initial sighting, the IEPA received in leader recreation of 8. The public notified of the potential for toxins in the water. The USGS and IEPA continued to collect samples until there was a shift in weather and cooler temperatures and rainfall seemingly moved the cyanohab out of the area. On June 28th, the IEPA finally received confirmation that cyanotoxins were no longer present in samples. This is a map of the established USGS water quality sites in Illinois with the location of the starved rock gauge where the bloom occurred and the upstream and downstream gauges in the black box shown here. Here's a closer view of those same locations, starved rock being the center, Seneca upstream, and Henry downstream. And those same gauges are marked with white arrows on this satellite view. All 3 of these gauges have chlorophyll and phycocyanin fluorescence sensors that were deployed at the time of the cyanohab occurrence, and they do display concentrations on the web in near real. So when we look at the phycocyanin data from this period around June 21st, we are able to see in real time the increases in phycocyanin concentrations as the bloom traveled downstream. As you see, phycocyanin peaked upstream near Seneca first, which is in the red, then starved rock, and finally at the downstream gauge near Henry. These continuous water quality monitors will continue to be used to identify conditions that may improve advanced warning of cyanohab occurrences. And although not all of the sample data from this event has been received and reviewed, there were some interesting preliminary results. Samples that were collected where the bloom was visible ranged from 14 to over 200 micrograms per liter, which were well above the recreational value. But samples that were collected where the bloom was not visible, while the microsystems were determined, the concentrations were below the recommended recreational level. And microsystems were the only cyanotoxins that were detected in USGS samples. 2 of the 5 and odor compounds that were sampled were detected. These are generally the most common taste and odor compounds. MIB was detected in all samples, and geosmin was detected in all but 1 sample. There are reports that indicate that humans can detect these taste and odor compounds at concentrations around 5 to 10 nanograms per liter. The MIB concentrations exceeded 5 nanograms per liter in all samples, but were greatest where the blooms were visible. Moving forward into 2022, samples will continue to be collected and current sensors will be maintained. Also, multispectral cameras will be installed at Starved Rock, and a mobile unit will be developed to use non-established sites. A nutrient diffusing substrate project will be implemented to explore which nutrients are potentially limiting algae growth. This will also include samples to develop a stream metabolism model, and new technologies will continue to be explored, including the use of eDNA analyzers. Obviously, with a project this size, there are many acknowledgements to be made, and there are a lot of folks involved. We will continue to add to this list of acknowledgements as we continue this project. And I want to thank everyone for your time, and hopefully you were able to hear most of what I said.
Yeah, thank you, Heather. And if you would be so kind as to stop sharing your screen, it would be appreciated. So as we move into the break, I once again want to remind everybody to use that Q&A feature in the Zoom toolbar to ask your questions. Specify a speaker, please. If the question is appropriate to specify to a speaker, we'll address them all in a question and answer panel with all of our speakers at the end of the morning session and before we break for lunch. Also, I noticed that there's a little bit of a bias going on in that those speakers who spoke first with their questions at the top of the list, thanks to upvotes, tend to get the most upvotes because they tend to be most seen. So be certain to scroll down to the bottom of the question list to peruse all the questions and upvote those that you find interesting, that you feel need to be most addressed. Your upvote will move the question up in priority, and we will, in the interest of time, because we won't have time to get to them all certainly, we will use your prioritization in order to prioritize how we ask those questions. There was something else I wanted to say about that, and I forgot. Oh, those of you who are speaking, I noticed that some of you are answering those questions in type. However, if we can, we'd like to have them addressed verbally, because there will be a recording of the session posted, and I'd like to have all subsequent viewers of the session be able to have access to those questions. That said, we won't get to all the questions, so if you are available to do so, speakers, I welcome you to stick around for a little bit after the session and take some time to type answers to those relevant questions that you have the time to address in type. Is there anything else you can think of, Amy, before we move to our break?
No, I think you covered it, and we will resume at 10:05. Excellent.
So we'll, as Amy says, we'll begin promptly at 10:05. You're in luck. I just checked. There's no line at the bathroom. So I look forward to seeing you all again at 10:05. Thank you for joining us today, and especially thank you to our presenters and to our symposium sponsors that you will see on our screen. Thank you. Welcome back, friends. We will launch right into it unless Amy has any housekeeping items before we do. Thank you, Amy. Next, I'd like to introduce, are you present and ready to fly, Hunter?
I'm here.
Fantastic. I'd like to then introduce Hunter Adams. Hunter is a laboratory supervisor, deputy quality control manager, and technical manager of microbiology and inorganic chemistry for the Cypress Environmental Laboratory for the city of Wichita Falls, Texas. You can read more of his outstanding biography on your screen, but without further ado, I'd like to introduce Hunter and his presentation. He will be speaking on proactive monitoring for HABs, taste and odor compounds, and cyanotoxins. Take it away, Hunter. You're welcome to share your screen as Amy unshares hers.
All right. Let me get this going. Does that look okay?
Looks good.
All right, so we've had a couple great presentations so far, and it's great to be participating in this research symposium today. My name's Hunter Adams, and I'm the laboratory supervisor for the city of Wichita Falls. We're a municipal laboratory owned and operated by the city in the public water system. So I want to start by thanking the National Institutes for Water Resources and the North Central Regional Water Network for the chance to spend some time talking about what we do in our lab today. And just for reference, Texas is a big state. We're in North Texas, just south of the Oklahoma border, where the star is on that logo. And today I'm really just here to tell you a story. It's a story of something that occurred in our water system for decades and wasn't really a problem for a long time, but it became a problem about 5 years ago. And then we're going to talk about what we did to mitigate that problem. So this is me, top right, and it's a pretty accurate representation of me when I start talking about algae and cyanobacteria. I tend to get a little excited, but that's good because that's why we're all here today. So I'm going to start by giving you an overview of what we use in our toolkit, and then we'll unpack each step on the following slides. SONs are a great starting point. They can be deployed, and the user can evaluate the data and determine when samples need to be collected based on parameters like pigment concentrations, dissolved oxygen, pH, temperature, maybe turbidity. Once we collect samples based on the SON data, we run counts using the flow cam. And we know cyanobacteria can produce taste and odor compounds and cyanotoxins, while the algae are really just problematic for the taste and odor compounds in fresh waters. So we need to know what's in the water to decide what to do next. From there, we can take two different routes. The first one is the taste and odor route. If we have taste and odor algae or cyanobacteria present in the samples, we start by running a modified threshold odor number flavor profile analysis test, which we'll talk more about in a little bit. And from there, we can try to fingerprint and detect and quantify the taste, odor, compound presence with a GCMS ECD system. For the toxin route, if we have cyanobacteria present, we have the possibility of cyanotoxins, obviously. So we need to know what's in there, and we want to run a quick qPCR screen to determine if the toxin producing genes are present. Then from there, we can either run a semi-quantitative Abraxas dipstick kit for confirmation, or send samples out to be run by LCMS, MS, or ELISA. And from what I've seen so far, we've been doing this about 6 years. Each step really complements and points toward the next, but no step by itself is standalone. So to explain where all this came from, because it's not common for a municipality to have this kind of capability, I want to start with a little background. The city of Wichita Falls is a surface water system. We're served by 3 source lakes. And from the 1960s, when the lakes were impounded, we've always experienced regular taste and odor cycles. Now, these cycles have always been a problem, but they were accepted as normal in our community. Everyone knew that several times a year, the tap water would smell and taste like dirt or grass or fish, or all 3 at the same time. Now, everything changed for us on July 7, 2014, when we began operation of the world's largest direct potable reuse system. Our combined lake levels had dropped below 20% due to historic drought, and the Texas Commission on Environmental Quality fast-tracked the permitting process for our emergency DPR. Now, in this, we piped 7.5 million gallons a day of wastewater effluent 12 miles above ground across town to a microfiltration reverse osmosis plant. And from there, we blended 5 mgD RO permeate with 5 mgD raw surface water and treated that blend in a conventional treatment plant. So that water was really treated more than 4 times in the process. And from this period from July 7, 2014 to July 21st, 2015, we didn't receive a single taste and odor complaint, which had never happened before. So our DPR was decommissioned in 2015 after record rainfall brought all 3 lakes to 100% in less than 3 weeks. And life went back to normal for a while until we had a couple TNO events in 2016. And the problem with this is we'd proved that we could go over a year without a taste and odor event, and the public did not respond well. The media came out, they asked us what was going on. We explained it in depth, told them what we were seeing, and they did what they do. They didn't listen to us and reported what they wanted to report anyway. So looking at this Genesis event for us, the first taste and odor event in 2016 after our DPR was decommissioned was a mix of algae, and we saw geosmin levels above 400 nanograms per liter in source and tap. It lasted over a week, and we received about 5 complaints a day. So by the time we realized what was happening with this and decided what to do in the water system, it was over. The second taste and odor event in 2016 was caused by an Anabina, Spermum bloom. And this produced MIB over 1,700 nanograms per liter in the source and 250 nanograms per liter in the tap. It lasted about 3 weeks, and I received hundreds of complaints. And if you're familiar with geosmin and MIB can be much more potent, and that is a lot of MIB in the tap water. So people were not happy. But thankfully, I wasn't the only person receiving complaint calls. The city council members also received a lot of complaint calls during that event. And they charged us with putting together a plan to prevent this from happening again in our water system. So this really brings us to present day when we're over 5 years into our monitoring program with 0 complaints from undetected taste and odor events. We have had a few that got past us, but we knew they were there, and we maxed out our mitigation, which we'll talk about in a little bit. And we've come a long way. The smellometer from the news headline on the earlier slide is their interpretation of a GCMS ECD system. And this picture in the middle on the bottom shows what our previous monitoring program really solely consisted of. A threshold odor number where we really just heated the water to 60 degrees Celsius and smelled it to see if it smelled or not. And that was basically all we were doing. And now we have a much more robust system, and that's me on the right with the nose. So before we get into more specifics, I just wanted to highlight some key differences in algae and cyanobacteria. The 2 presenters have already done some of this, but I wanted to throw this up there. I know there's a lot on the slide, but you can pull this, you can take a screenshot or take it from the PDF that'll be provided after the presentations. And I wanted to point out that term algae is frequently used to describe both groups. but each are unique. Algae are protists, cyanobacteria are actually true bacteria. Cyanobacteria produce the pigment phycocyanin, while algae don't. Algae can be TNO producers, but only cyanobacteria produce cyanotoxins. Algae produce toxins, but not in fresh waters known to affect humans. So we're not as worried about those in that respect in those areas. So at least for us in our area, cyanobacteria are really our main nuisance organism, but algae can be problematic as well. So we look for both. Just want to understand there's a distinct difference in these two types of organisms, but the term algae is normally used as a catch-all. So a new approach, how did we get to this point with our monitoring program? And then we're going to look at the different steps in our monitoring program. We had to set up a list of questions to follow to guide what we were going to do. What did we want to accomplish with this program? So first, we need to know if algae and cyanobacteria are present in the water. Then we need to know how many are present. We know that blooms exemplify the remarkable ability of these organisms to really exploit their environment. They're symptomatic of some degree of ecosystem imbalance. And we know they respond to broad environmental stimuli like light and temperature and nutrients. And I'm in North Texas, so it's 20 degrees today, but that's not common. We normally have plenty of light, temperature, and nutrients. So next we needed to know, can the organisms produce taste and odor compounds and or cyanotoxins? They're both metabolites. TNO compounds are VOCs that are non-toxic, while we know the cyanobacteria can produce the toxins that can affect humans and wildlife. And to decide the next steps, we need to know the TNO concentration and the toxin concentrations that are present in the waters. And to answer that question, we put together an integrated approach. So we took pieces from our micro lab and pieces from our analytical lab. We merged those and we use combined results that help us respond to an event. So one of the first things we were already doing, but maybe not doing well, was monitoring with YSI EX01 sondes. So we were doing thermal profiling, taking temp readings and DO readings every foot of depth to detect stratification. Now we've got an aeration system at both lake intakes, and we normally don't see any stratification when it's operating correctly. So our profiling has helped us detect failures in the aeration system when it has stopped and nobody from our maintenance division has noticed it. And it's helped us see when a thermocline is starting to form before it actually happens. We can see it when it's on the way. We've also recently started using a YSI total algae sensor to track chlorophyll A and phycocyanin pigment concentrations. And we can use those ratios to determine if the cell counts we do in our micro lab align. Now, we also do sampling on the lakes. We profile every foot of depth with the sonde, looking at things like temp, pH, DO, ORP, and pigments. We also look for nutrients and nitrogen species and phosphorus. With a Van Doren sampler, we take samples every 3 foot of depth on 3 different locations in the lake, upstream, at the intake, and we have an outfall where our reuse system is now permanent. So we put all of our wastewater effluent back into one of our source lakes, which you know can be problematic because we do phosphorus removal at the wastewater treatment plants, but the engineers told the city that nitrogen will not be a problem. And if you know much about algae and cyanobacteria, that's not exactly right. So we do basically surface sampling as well, and we run everything we can run in our analytical lab and our micro labs on it just to get a good overall water quality picture. The next piece of our plan that we were already doing was algae and cyanobacteria identification by light microscopy. Now, this method can take hours, and it wasn't practical for us. There will always be a need for expert taxonomists that can ID organisms down to the species level. But for a water system that needs to react quickly like us, that wasn't practical for us. We didn't have that kind of expertise, and we didn't have that kind of time that needs to be devoted to a project like that. So we added a flow cam to our toolkit. This is a semi-automated instrument that images and enumerates algae and cyanobacteria into preset method categories. So we classify them into things like TNO producers, cyanotoxin producers, filter cloggers, green algae, detritus, and just the other junk in the sample. And the most significant advantage to this type of technology for us is runtime. It's short. And then I can open saved run samples to view previous sample batches. So it takes us about 3 minutes to process a sample and only a few more to sort. Now I can train new techs and interns on how to process a sample, how to run it, and save the file in the software. And then afterward, I can sit down with them. We can open the run file and I can do the classification with them until they're more comfortable doing it on their own. Or I can also do spot checks after they've been released to do this on their own to make sure they are in fact IDing things like they should be. So what you're seeing on this screen is what an analyst sees when a sample runs in real time. We use a 1 mil sample and images are captured and sorted into collages. Then the software enumerates the organisms based on sample volume. We use particles per mil as our unit to track and make treatment decisions. And we spent the first year running 5 days a week, establishing a baseline of what we see in our water system, because we wanted to be able to establish triggers on when to do more investigation. So because of that, now we know that Anabina or Dolichospermum in excess of 200 per mil, it's indicative of a problem for us. While if we see something like Pediastrum, really no matter how much of this we've seen in our water system, it's not an issue. We also see organisms like the dinoflagellate Peridinium and Aphanizomenon, which is a type of cyanobacteria that can both be problematic for us. But we've seen that Peridinium isn't really an issue unless it reaches around 500 to 600 per mil in our water system, while Aphanizomenon maybe around 300 per mil. So the flow cam has allowed us to quickly look at trends on a more specific level instead of viewing them as a total algae count. But we still keep microscopes serviced and on hand to do manual counts and identification as needed. So the next piece of our plan, and this was the new part, this is the smellometer that the news talked about. It's a TNO monitoring quantification by a GCMS ECD system. And we use the taste, odor, wheel, and standard methods, which categorizes and identifies some known taste and odor compounds. We worked with Thermo Fisher Scientific to put together basically a Franken instrument. We wanted a system that could run methods for each category we felt would be an issue for us without having to really manually change columns and other components in between. So we're a large lab, but we don't have a huge number of staff. So we need everything to be as automated as possible. And we knew geosmin and MIB were an issue. So this is where we focused the majority of our time. For the earthy, musky, moldy category, we use a trace gold 5MS column and a DVB CAR PDMS SPME fiber for solid phase micro extraction. Now we also set up to run the grassy, hay, straw, woody category using the same column, same fiber, and same internal standard, which is IBMP. The fishy, rancid category is one that we knew we'd smelled in our surface water before, and we know it's produced by diatoms, dinoflagellates, and algae with high cellular polyunsaturated fatty acids. And these can be problematic for us. So we developed a method to run these on the same column, but using a DVB PDMS fiber. So when we bought this instrument, we bought a Triplus RSH auto sampler with a 3-position sample tool station. So no manual change out is needed in between. Now, this method involves a long derivatization step. So we run this one over the weekend or overnight. The last category we really wanted to focus on is the moldy, swampy, septic, sulfurous category. We do this by headspace using a trace gold 1MS column and an ECD as the detector. And we can also do the phenolic categories on the wheel and the hydrocarbon categories using existing instrumentation. In 2020, we published an article in LCGC North America, and this outlines the taste and odor categories we monitor. You can see on the right, it's a table of all the method parameters for our GCMS ECD system. So if you're interested in this, you can Google that or shoot me an email. I can send it to you, and they're all on ResearchGate. So this slide shows all the taste and odor events that we've identified since we began this monitoring program. We've been able to knock out most events that reach the distribution system, and even events with very high concentrations have, for the most part, been mitigated by source switching due to early detection. And you can see, looking at these numbers and dates, it's a problem really persistent in the warmer months, which is not surprising, but we have seen blooms in cooler months. You know, we're in Texas. It was 90 degrees on Christmas Day here, which was not a lot of fun. Now, keep in mind this geosmin level from April 2020. We're going to look at that on another slide. Then we've also had a few this year in the trichloroanisole, the second from the bottom, that was produced in filter media, so that was interesting. So by merging these technologies, we've been able to identify and correlate specific algae to taste and odor events. This is an example from June 2017, where you can see the Anabina spike matches with our geosmin levels we were seeing and how we achieved a kill with copper sulfate that resulted in lower counts and declining geosmin levels. Now, this is one of the ways we establish baselines for treatment triggers. And like I said, Anabina is one of our main cyanobacteria nuisance organisms. It can produce geosmin. It can produce some really potent MIB, and it can also produce saxitoxin. So it's one we keep an eye out for. We see a lot of, especially in the summers. Now, these graphs are from April 2020, when we had another, this time it was an Anabina explosion at Lake Arrowhead. Lake Arrowhead is normally brown. That's a picture in a filter housing that I had run that day. In this bloom, we saw concentration of geosmin over 11,000 nanograms per liter in a few days. And our mechanics went to treat the lake with copper sulfate. Now, you can see immediately after that, the Anabina counts declined basically immediately, while the geosmin level spiked to 14,000 nanograms per liter when the intracellular geosmin was released during the kill. Then the geosmin concentrations fell out to a non-detect within a few days. And this is one of those events where you could smell the lake before you could ever see the lake when you were driving up to it. It's not a fun time to work for a water system, but it's a fun time to be in the lab. The next major piece of our toolkit is qPCR. This is a molecular-based test that we use as a screening tool for cyanotoxin-producing genes. We use EPA 705.0 and phytoxygen cyano-detect kits to monitor both surface reservoirs and our secondary reservoir. This kit's standardized so we can detect and quantify these cyanotoxin-producing genes. It takes us about 60 minutes to prep samples and another 70 or so runtime on a Thermo Studio 5 thermocycler. It's important to note with this that the test detects the presence of the genes, not the toxins themselves. And we've seen a few low-level detections by qPCR. In July 2018 and July 2019, we detected the presence of the microcystin nodularis gene cluster. And in both of these events, qPCR detection was below the lowest calibration standard, but we still sent samples off for confirmation, which will be shown on another slide. It's also interesting to note here that we had similar populations of cyanobacteria present during both events. Now, in the last few years, we've also recently started using Abraxas dipstick kits as an intermediary between qPCR and sending samples out for analytical confirmation. These tests are semi-quantitative, meaning they'll let you know if the toxin is present and give you an idea of the concentration. They're fast and they're cost-effective. We use them just to see what we need to do based on what we're seeing by qPCR and counts with the flow cam. The jury's really still out how effective these are for us, but we don't have existing analytical instrumentation to do ELISA or LC-MS testing. So it's just something we keep on hand to use as needed. So qPCR, that allows us to screen samples quickly, and the dipsticks allow us to decide if we need to move forward sending samples out for confirmation. And like I said, we don't currently have the instrumentation needed for analytical confirmation or biochemical confirmation. And we send samples to Eurofenazine Analytical for EPA 545 and 546 as needed based on the results we get from the other two tests. And in 2018, the toxins were confirmed at low levels in our source and one water treatment plant. Now, Cypress Water Treatment Plant has the MFRO, the microfiltration reverse osmosis plant. So we thought this was interesting because we believe that the toxins were likely present but blended out in the MFRO. Jasper Water Treatment Plant is just a conventional treatment plant. And we currently only send samples off for microcystin and cylindrous remotin detections based on EPA health advisory limits. So just to summarize our monitoring program, what we've talked about so far, we adjust our frequency seasonally. We run more frequently in warmer months, obviously, and less in cooler, but they are subject to change by annual review or triggers as needed. We run algae counts by flow cam. We run the modified TON FPA where we still heat the water up, but this time we're looking at it, we want to know how smelly the water is and what type of smell it has. Is it earthy? That tells me what GC category to run. Is it sulfurous? That tells me what GC category to run. So we use that as to provide some direction for us. We monitor actinomycetes. They're fungal-like bacteria that can produce a host of compounds like geosmin, MIB, halogenated anissols, pyrazines, and they can become problematic in the distribution system. You know, they're ubiquitous in soils. You find a lot of them in surface water, especially ours with a lot of sediment, and they can be problematic in filter media and in distribution systems. So we keep our eye on those selected ion monitoring for our most problematic category, the earthy, musty, moldy, and we run scans for the others. And then we do weekly qPCR and Abraxas dipsticks as needed to help determine the need to send samples out for confirmation. Now, the triggers we established in the first years, they've been adjusted over time. If counts hit trigger levels depending on the type of algae, we can switch source lakes or treat the lake with copper sulfate and citric acid to kill the bloom. Now, the citric acid helps increase the solubility of the copper by lowering the pH of the source water. We only do this if it's triggered at both lakes, because the kill is going to result in the release of the intracellular taste and odor compounds or cyanotoxins, and they're not as easily removed as if you remove them while they're still contained in the cells at the treatment plants. High TONs or FPAs detect additional scans. Levels of geosmin or MIB over 10 nanograms per liter trigger daily testing, because the human detection limit is somewhere around 10 part per trillion. Customer complaints, if we receive any, that would trigger additional testing. Then we also monitor the distribution system. We use our revised total coliform rule, BACT sites, and nitrification action plan sites that state of Texas requires us to keep. We use those because we're there daily anyway. And then we do flushing as needed if the problem is localized. Now, at the water treatment plants, we run potassium permanganate year-round in recovery wells. And during a TNO event, we add this at the clarifier weirs as well at 0.4 milligrams per liter. We use powdered activated carbon based on GCMS results. And we started doing this in 2016 based on Paul Westerhoff's PAC curve he published. Excuse me. Now, when we started this, we maxed out at 100 milligrams per liter because we didn't really know what we were doing at the time. And it turned our clarifier effluent black. And as the name suggests, it should be clarified effluent. It should be pretty clear. So we don't do that anymore. We learned our lesson with that. In the event we exhaust all treatment options, we know there's a bloom, we're doing whatever we can in the treatment plants. We have a template press release ready to go because we want to notify the public the extent of the bloom, the steps we've taken to mitigate the effects of the bloom, and just to reassure them that we're actively working on the issue. Now, it's a collaborative effort between administrators in our public works department. Now, on this slide, I've got some publications that have come out in our lab in the last few years on taste and odor production. They primarily cover algae and cyanobacteria monitoring. But as you can see with the one on the filter right, we've also done some work detecting taste and odor compounds in filter media. And this monitoring program for algae and cyanobacteria is what alerted us to that problem. So that was pretty cool. We also have a few more in review that are going to come out soon. Here I've got some guidance documents and publications that we've been involved in that may be of interest to you. Our taste and odor guidance manual was updated in 2021. It's available online. The ITRC guidance document for harmful cyanobacterial blooms came out in 2021. We also have another in progress that focuses on benthic harmful cyanobacterial blooms that should come out in the spring of this year. So I'm excited about that. APHL's cyanotoxin guidance document for laboratories came out in 2021. It's a really good review. If you're interested, Google that. AWWA's M57. Now, this is the first edition. We are in progress with the second edition. It's in the peer review phase. It's got a lot of updates, some really good taxonomic keys, updated taxonomy, a chapter on molecular methods, satellite imagery, all kinds of new things, a big chapter on taste and odor compounds and cyanotoxin production. So we're really excited for this to come out. And then in 2019, we helped with a pictorial guide for FlowCam users. So just to summarize, science works, and thankfully it's been effective to this point for us. Again, we've received 0 complaints for 1,914 consecutive days from an event that went undetected in our water system. Like I said, we've had a few that occurred, and we did detect and maxed out treatment, but some taste and odor remained in our finished tap water. And because of this, after 2 years, the news called and said, something's changed. The water doesn't smell and taste bad anymore. So they came out, they did a nice story on what we were doing. And then Francis Perkins, who worked for Yokogawa Fluid Imaging Technologies at the time, encouraged us to start talking about this story, to get it out there. We ran a short article in OpFlow in 2018. And since then, we've had the opportunity to present all over the country and help other utilities that are facing this issue and other labs and people interested in this kind of work with things we've already dealt with. And we have another article coming out in June 2021 on a June 2021 bloom coming out in the May journal AWWA. So we're excited about that. That's going to present some data on what we saw in all the different pieces of this monitoring program. So lastly, this program wouldn't be possible without our plant operators and our plant mechanics. They make the changes when we tell them triggers have been hit. The co-authors listed here, they've been instrumental getting this program going and keeping it going. And this type of monitoring is not easy. It's not cheap. It takes time and it takes patience, but it can be very effective when modified for individual systems. And it's kind of a fluid monitoring program. So the focus can quickly shift between organisms, parameters, taste and odor compounds, and or cyanotoxins, depending on what the early warning signs indicate in your waters. With that, that concludes my presentation, and I'll be happy to take any questions by email or during the Q&A portion at the end of this session this morning. Thanks.
Excellent. Thank you so much, Hunter. And once again, I'd like to remind our attendees to indeed use the Q&A feature in the Zoom toolbar to type your questions. Specify a speaker, please, if appropriate. And also, we will prioritize the questions asked during the panel by upvote. So upvote those questions that are already posted, and don't forget to scroll to the bottom of the page of questions and upvote those recently asked questions as well. Otherwise, we'll end up favoring the person who just happened to speak the longest ago. So once again, thanks, Hunter, for a great presentation. And I'd like to introduce our next great presenter, Shui Ma. Shui is currently a third-year PhD student in natural resources and environmental sciences at University of Illinois, Urbana-Champaign. And you can read the rest of his bio here, of course. If you are ready, Shui, I'd love to hear your presentation. Interaction of hydrological and anthropogenic processes controls the relationship between stream flow discharge and nitrogen concentration in the U.S. Midwestern watersheds. Are you ready, Shui?
Yeah, I will share the screen. Thanks for the introduction. And good morning, everyone. And as the introduction, this is Zoe, and I'm now a PhD student at the U of I in NGWAS lab. And thanks for giving the opportunity to share our study. And here we make the title more simplified. Yeah, here as agricultural nitrate export pattern shaped by crop rotation and tile drainage. And I would thank all the collaborators in this work. And in our work mainly focus on the nitrogen export patterns in the agricultural watershed. And as we all know, nitrate is an important nutrient that stimulates the harmful algal blooms there. And yeah, this is the content for today's sharing, including the introduction to nitrate dynamics in the US Midwest, and then a proposed mechanism, hydrological vertical mixing, which is used to explain the observed export patterns. Then we will dive into the seasonal and spatial pattern of the nitrogen exporting pattern over the watershed. And we also made a prediction over the central US Midwest at the HAB 8 watershed based on the developed model. And the last part is the conclusion part.
Before you move on, Shui, there's been a request that you speak a little more loudly, that you seem a little muffled. So if you don't mind speaking a little loudly, please do.
Okay, yeah.
Thank you.
This is a map shows the area in the United States that were glaciated. Basically, the colored region here. Oh, sorry, the colored region here, and from that figure, we could know that the U.S. Midwest was once glaciated in this period, and the glacial material formed a below-ground impermeable layer in many places, and it's one of the major reasons for the poor drainage conditions there. And then the tall grasses and forests dominated this region. And here is the following picture, and humorous poem suggests that how pioneers view the wet prairie and the malaria associated with it. And because of the tall grasses and forest, the organic matter from the leaf falls in forest, and the root growth in grassland build up fertile soil with high agricultural capacity there, and which makes the US Midwest one of the fertile regions in the world. And in the last hundreds years, the region with fertile soil and poor drainage condition has been converted to agricultural land. The artificial drainage, both the tile drainage and the surface drainage, were widely adapted to improve the drainage condition in this region. And as we all know, tile drainage flow is a major source of nitrate in the agricultural watershed. And here the left panel shows the area-wise tile drainage fraction in the different HAB 8 watershed in the central US Midwest, and mainly located in 4 states: Iowa, Illinois, Indiana, and Ohio. And the right 2 panels shows 9 years average corn soybean fraction in different watersheds, indicating the development of agricultural production in this region. Yeah, I'm not sure whether you all know what is tile drainage here, and I put a figure here shows the drain system. Basically, here it shows the open ditches corresponding to the surface drainage, and the subsurface drainage system corresponding to the tile drainage there, and it's widely adapted in the US Midwest. And with the development of human society, lots of environmental issues emerge. The development drainage system exacerbates floodings, and the emission from both point and non-point sources leads to the aquatic pollutions. And one of them is that the excessive nitrogen from the agricultural landscape flows along the Mississippi River contribute to the formation of dead zone in the harmful algal blooms in the Gulf of Mexico. Thus, understanding how nitrogen dynamics interact with hydrology in agriculture watersheds is critical to develop effective policy in nutrient loss reduction. And this is what we are trying to tackle in this study. Yeah. And a common perception is that nitrogen export from agricultural land typically follows a chemostatic or biogeochemical stationary pattern, where solute concentration that varies little across wide range of the discharge. As the upper panel here shows, the nitrogen concentration almost keeps constant in different years. However, we do observe the variance in discharge. And the recent study has revealed nitrogen concentration and discharge relationship from agriculture landscape mostly follow a flashing pattern here, like this one. From example from EU watershed, the solute concentration increase with the increase of discharge here. And shallow and deep hypothesis has developed to explain the flashing pattern. However, the predominant factors that shapes the nitrogen variation and export pattern are still missing. So in this study, we downloaded the data from USGS gas station from 82 watershed across the US Midwest. And we found that the nitrogen concentration, nitrate concentrate C and discharge Q here relationship show a distinct pattern here. Like this one, the nitrate concentration increase with the increase of discharge and the low flow stage, and then reach a plateau there. And in some watershed, nitrate concentration will decrease, like this one, will decrease with the increase of discharge and a very high discharge condition, like this one here. And based on that, also I need to mention that different colors in the figure indicate the data point collected in different months, and we do see a seasonal variation of those patterns. And based on the observations, we want to ask the following questions. The first one is, what are the underlying processes of the CQ relationship, and how do they shape the distinct CQ relationship in the US Midwest? And what factors control the seasonal interannual variability of the CQ relationship, and how do different watershed characteristics. Here, the cone fraction and time drainage fraction affect the spatial variability of such nitrate export patterns here. And inspired by the shallow and deep hypothesis, we further hypothesize that the hydrological vertical mixing of quick flow here and the slow flow is what shapes the distinct circular relationship observed in agricultural watershed. And from this schematic figure, we could know the quick flow and the slow flow has different passes, which shapes the distinct characteristics of the nitrate concentration there. And here we define the quick flow shown here as the flow parts that respond quickly to the rainfall event. And the major source of quick flow include surface flow, shallow subsurface flow, and the tile drainage flow there. And the nitrogen concentration in quick flow is generally high and also dynamics in agricultural fields due to the superficial nitrate input of nitrogen fertilizer and seasonality of agricultural practice. And the slow flow refers to the flow path that responds slowly to rainfall events here in this figure. And the major source of the slow flow include deep subsurface water and groundwater. And the nitrate concentration is slow flow is generally low. And based on the reasoning, we developed a parsimonious two-end member mathematical model to illustrate the mixing process of quick flow and slow flow. The mixing model can be divided into two parts here. One is a hydrology part and another one is a biogeochemical part. And in terms of the hydrology part, we use such simple assumptions and equations to simulate the hydrology dynamics in the quick flow and the slow flow. And the stream flow is the mixing of quick flow and the slow flow here. And then this is a biogeochemical part. The stream nitrate concentration is a weighted average of quick flow and slow flow. For simplicity here, we assume that nitrate concentration follows a constant changing rate during a rainfall event. And here is an example from the Spoon River watershed in Illinois. We firstly identified a rainfall event from the hydrograph, like showing the same region here. And this is the detailed information about the time series of both discharge and nitrate concentration. And we used the above mentioned method to develop the circular relationship. And the red line here is what we got based on our model, and the blue dot is what we got directly from the observations. And the result shows the proposed hydrological vertical mixing model can successfully reproduce a circular relationship and a single rainfall event. And we could know from the model that a flashing pattern and low flow region indicator transition from a slow flow dominant flow to a high flow dominant flow. And the plateau region indicate a relative stable quick flow concentration there. And from previous slides, we have seen that circular relationship has shown a seasonal variation, and we hypothesize that the seasonal variation of agricultural practices and the annual cycle of different seasons are the drivers to that. And to investigate how seasonal variation of biogeochemical factor and hydrological factors that drives the seasonal variation of the circular relationship, we here construct a monthly conceptualized rainfall event for each month with the monthly statistics derived from historical hydrograph and observe the stream nitrate concentration. And the basic idea here is to use a representative rainfall event like showing the right columns to represent all the rainfall events happens in a certain month there. what we need to do is to derive all the parameters in the conceptualized rainfall event here. And and I will show you what we have done here. The first step is to identify the slow flow part using a digital filter. And here's what we get from what we get. The red line actually shows the slow responded flow parts there. The magnitude is small and the variation is small. So another part is based on the digital filter, we can also get the quickly responded discharge here corresponding to the what we show in the blue line here. And this part responds quickly to rainfall event, and you could find a spike like this one and this one. And this once we get a slowly responded part, we could simply assume estimated slow flow part from the slowly responded discharge we get from the previous step. And then we here shows we extracted all the rainfall events that happens in a chosen month from the hydrograph after applying a 3 days moving average field, as shown here. And the highlighted region indicate different detected rainfall events, and this is the detailed information for different detected rainfall events. And we estimated the flow recession rate of the conceptualized rainfall event as average flow recession rate of the detected rainfall event in a certain month here. And with all the parameters we get, we now can construct a conceptualized rainfall event like this one to represent represent hydrological processes of the rainfall event during a certain month here. Yeah, then we will have the biogeochemical part. For the quick flow part, we characteristic the quick flow with initial quick flow and the changing rate there. And the initial quick flow is estimated with nitrogen concentration of the top discharge part. And we assumed we get the changing rate of the quick flow concentration by taking the gradient of the initial quick flow there, and the slow and the nitrogen concentration in the slow flow is estimated with the nitrogen concentration of the lowest 5th percentile discharge there. And based on the averaging of the observed pattern in the 82 watershed in the US Midwest and the developed a model, we firstly find a season variation of quick flow largely follows a temporal pattern of field level farmland nitrogen dynamics there, which supports the hydrological vertical mixing theory. And in particularly, the quick flow concentration increase here from March to June corresponding to the synthetic fertilizer application for corn, and increased the soil mineralization during the wet and warming up period. And and the increasing of quick flow concentration would move the cycle relationship upwards in the cycle relationship here. And then the decrease of nitrogen concentration from June to August corresponding to the actively plant uptake in this period, and the decrease of quick flow concentration actually moves the cycle relationship downwards in this figure. And from the September to November, the quick flow concentration slowly increased due to the mineralization of soil organic nitrogen there. And similarly, that would cause this circulation shape to move upwards during this period. And from November to February, due to the low microbial activities, the sea creek remains relatively stable, and the circulation shape also remains relatively stable. And the watershed integrated all the findings, and we can know that the watershed scale pattern closely mimic the field level observations there. So we checked how watershed characteristics, as I said before, the cone fraction and the tile fraction would affect the circular relationship, especially especially in June, which is one of the months with the highest nitrate concentration and flow discharge, and therefore the highest nitrogen loading is there. And from this figure, we could know that the quick flow concentration increases exponentially with the increase of cone fractions there, indicate a large portion of stream nitrate originates from fertilizer application in cornfields. In this figure, we could know that the slow flow in June is also significantly correlated with cone fraction. And we explain this by the reason that evapotranspiration of cropland is generally low in May and June compared to that in forest and grassland, as the relative low ETU, the relative low evapotranspiration will lead to more water storage in June, which explains why Q slow is higher in the watershed with higher cone fraction there. Other critical variables like the stow flow parts and the stow flow concentration has shown no significant correlations or weak correlation with cone fraction. And based on those empirical relationship we observed from the observed data, we integrated those into the previous developed hydrological mixing model. We could find that the increasing of cone fraction would move the circulation shape top right corner. And the nitrate concentration remains a relatively stable value as a change of stream flow with a cone fraction of 0.1 here or 0.5 here, and indicates that the observed distinct two-stage relationship might be a unique feature in the agricultural landscapes. And similarly, we find tile drainage is significantly modified hydrological process and further influence the biogeochemical cycle in the agricultural systems in July. Similarly, the sea creek is significantly correlated with tile fraction across all watershed, and it's higher in watershed with more tile drainage fields. The relationship can be explained largely by decreasing residence time and less nitrogen absorption by soil in tile drainage field. And here we also find the slow flow concentration decreases exponentially with the increase of tile fraction here, suggesting that slow flow might originate from deeper soil with lower nitrogen concentration. We'll also find high flow in June has a positive high flow part in June has a positive correlation with tile drainage fraction there, and Q slow in June and the low Q slow there and Q slow here and the low flow part are negatively correlated with tile fraction, suggesting that tile drainage would facilitate quick flow drainage and reduce the water penetration that contribute to the deep slower flow. And integrated all the results, we could find the increase of tile drainage fraction here would move the circular relationship towards the top left corner here. And here we will briefly go through how tile drainage and core fraction would impact circulation in different months. And the 12 figures shows the relationship between the quick flow concentration and core fraction in different months. And we found that the significant correlations between quick flow concentration and core fraction in different months. And this pattern may arise from both the short-term and long-term legacy effects of hydrological and biogeochemical cycles. And we will not dive into the detail about that. And this figure shows how the core fraction, the relationship between the core fraction and the slow flow part, and we do observe the seasonal variation there, and we can see a huge impact of on slow flow part of core fraction in June and October, corresponding to the low ET period, evapotranspiration period of the crop fields there. And the tile fraction, from those figure, we can see that the tile drain also have this negative correlations between the slow flow part, suggesting that tile drainage would decrease the watershed storage and lower the water table in the watershed, and this is uniform in different months. And the impact of tile fraction on the high part of the flow shows the seasonalities, and this is and the tile drainage may play a more important role in relative wet period, like in May and June, and also due to the melting of the snow, we also observed a positive and significant relationship in those months. However, in June and August and September, also August corresponding to the dry period as well as the active uptake of plants, the correlation is not so good compared with other months. And the impact of tile fraction of quick flow concentration also shows a seasonality, and it's also more important in months with more water sources and more available nitrogen. Integrating all of those findings, we developed an empirical model to combine the hydrological and biogeochemical effects that predict quick flow concentration and assess the model performance through leaf-1 watershed out cross-validation there. And those figures shows cross-validation result in different months. And the R-square in cross-validation ranges from 0.25 to 0.64. And overall, the model performs well in prediction nitrogen concentration. And here's the result for the cross-validation for the slow flow concentration part. And based on the developed model, we could use various decomposition methods to attribute the variation of quick flow concentration across the selected watershed into the spatial variation of core fraction and tile fraction here. And we find that the core fraction explains nearly half of the variance of quick flow concentration in the quick flow concentration in June, and the tile fraction contributes around a quarter of the variance of quick flow there. And then we applied the model and the HAB 8 watershed in the central US Midwest. The result here we show the result for 3 key months, and the concentration prediction model successfully reproduced the seasonality of quick flow concentration, which is the quick flow concentration decreasing from June to August, and increased from August to November, corresponding to different agricultural practices there. And the spatial pattern agree with well with existing riverine nitrate loading prediction model in previous study. High concentration in most part of Iowa and central Illinois and Indiana. With a parameter related to nitrate concentration, we also introduced the daily discharge data from the GRACE dataset, in which case we could now construct secure relationship in many watersheds and calculate the nitrogen loading, which is the product of a concentration and discharge. Based on that, we performed a simple scenario analysis here, and we increase the tile drainage fraction for different percentage here. And we found that increased the spring weighting because as the fact that the increased spring weighting has been projected for the US Midwest, and which may lead to more tile drainage there. And our results suggested that increase of tile drainage fraction would increase the nitrogen loading for all of the HAB 8 watershed in the central US Midwest here. Like in this one, the middle column is the absolute nitrogen loading increasing here. This is the increasing is especially the case in the northeast Indiana and northern Ohio, where 30% relative increase of tile fracturing on the current condition will lead to 20% more nitrate loading in June, and larger nitrate loading already occurs in the watershed with high tile fractions, highlighting the importance of nitrogen reduction in terms of the tile drainage there. And here's a conclusion part of our work. Our result indicates that hydrological vertical mixing is the dominant factor that shapes the distinct secure relationship in the US Midwest. And the chemostatic or chemodynamics behavior of nitrate are associated with low flow status and their sources. The distinct relationship in the central US Midwest is shaped by its unique landscape and practices. And the seasonal variation of the secure relationship reveals the seasonality of watershed hydrology and biogeochemical properties and how they interact with each other. The hydrological mixing model can be further extended to other agricultural watersheds all over the world with higher nitrogen concentration on the surface soil layer due to the anthropogenic fertilizer application. And the model for the first time quantifies the role of cone fraction and tile fraction in explaining the spatial variation on quick flow concentrations there, highlighting the role of tile drainage in regulating nitrogen loss reduction there. And our work also provides a framework for future analysis of managed practice on nutrient loss reductions there. And those extreme nitrogen concentration characteristics support the hydrological vertical mixing. And we think it requires further testing to see if the underlying mechanisms are true if their serial holds in different locations. Thanks.
Thank you, Suwei. So now we will move into the question and answer panel. If you have any questions still outstanding, please type them to the question and answer feature in the Zoom toolbar. Scroll to the bottom to check all those questions. If you see a question that you'd really like to have addressed, please upvote it as quickly as possible because we probably won't see it if it's at the bottom of the list. Once again, any of the speakers in attendance, I would ask if we don't get to the questions that are there verbally, if you have a few minutes after the session to stay on and type some answers as a courtesy to the askers. If you're available to do so, please do so. However, we will certainly give preference to verbal questions and answers and frankly discussion, so that those can be featured in the subsequently posted recording. And I would like to consider this a panel. So if any of our speakers have additional insight along with the specifically asked presenter, please do make this a panel discussion. Before we move into that panel discussion, I'm also going to take another moment to again thank all of our sponsors that are now appearing on the screen. Amy, if you don't mind stopping the share so that as our presenters are asked questions, we can see the faces of our presenters as they are speaking. And quickly, before we move into that, since Michael asked, I have provided Michael with a delicious virtual lunch. Unfortunately, it's not very filling, so you might want to raid your own refrigerator to augment the virtual lunch that I provided for you, Michael. With that, let's move to our question and answer session. So the first question is addressed to Matt, and it comes from an entity called Lake Environmental. Matt, what do your field sampling kits look like? What's included and who is collecting samples? Are they volunteers?
Yeah, thanks for the question. So for those of you that were able to see the responses to that question, I'll just reiterate what Gina Liberty, who's with the Wisconsin Department of Natural Resources, since most of the bloom activity has been occurring within the Wisconsin shoreline of Lake Superior, the DNR coordinated the putting together these kits. And so basically what the kits were only available to our rapid response team members. So that's people that are one of the agencies involved in the bloom response or one of the universities. So for instance, our group at Northland College has one of the rapid response kits. So there was just a limited amount that were available, but they were forwarded by Gina to the rapid response team. They have bottles for algal ID, microcystin and other toxins, and then a glass vial for the saxitoxin analysis. So we're trying to get an idea of who's living there in the blooms and then were there toxins present. And then those samples were shipped to the Wisconsin State Lab of Hygiene in Madison for analysis. So that's what we had for our kits that everyone was using, but the response team was also that part of what we were doing there was trying to get people on the water with other kinds of sondes and water quality bottles just to sample and get as many different kind of parameters as possible. So it's kind of a combination of the kits that we did have prepared by DNR and then whatever bottles anybody else was able to grab and get analyzed as well. I don't know if I also have my co-authors, I believe Kate and Brenda are able to speak too. So I'm going to pass off some of the questions that may be for us to them, and if they want to add anything else, feel free to do so.
Great. Well, chime in at this moment if you do have something to add. Otherwise, at this time, I'll just say thanks, Matt, for this reply. This question is generic enough that I think it really applies to all of our morning speakers. Garrett asks what PPE, personal protective equipment, of course, is recommended when working around harmful algal blooms. So all speakers, feel free to offer something.
This is definitely not an area in my wheelhouse. So
I believe that question came through when Heather was presenting, and I'm not sure that she's still on.
Heather, I know, is having some internet troubles. So I mean, the biggest concerns are going to be related to ingestion. And ingestion can be incidental, you know, even by getting aerosols in your mucous membranes, right? So ordinarily, you're pretty safe to just take some kind of course of action that minimizes ingestion, including thoroughly washing yourself, because rubbing your nose, for example, can introduce some of the toxins if you've had your hands in the stuff. So gloves are a good idea. Some kind of face mask that prevents aerosols from getting into your mucus. But it's going to vary a lot by scenario as well, right? Whether you're in the field or in the lab. So look for the protocols that might be in place for your institution and adhere to them. That's kind of my off the cuff answer as a guy who spends a lot of time on water where stinky things occur. And if anybody wants to add to that, I'd be keen to hear it. If not, let's take Caitlin's question, who also addresses Matt. Caitlin's evidently from the Minneapolis Park and Recreation Board. Would you be willing to share the rapid bloom response communication plan?
I think I'll let Kate Reinl. Kate provided some response. She's the one who's been coordinating that. Kate?
Yeah, thanks, Matt. And great presentations, all of our presenters. I've actually gotten several emails already. So many of you are interested in kind of what we've organized for our bloom response plan. I won't want to take up too much time on here kind of going through the details of what that is. But if you do want to email me, I can provide all of those materials as well as an explanation of kind of how we organize things. But overall, we basically have this core group of people. We have an algal bloom subgroup. We meet about once a month. And those of us that have capabilities for sampling, basically every year we take an inventory of what resources we have available for rapid response. And we have kind of this template and phone tree that we follow. So there's kind of this trickle down effect. If someone sees a bloom, several people are notified, they disseminate that information to their labs and we all get out there. And we have a plan for who is able to collect what types of samples and analyze them after the event. And so we have kind of like a working document and a couple of tables that go along with that, as well as information on the kits that we use during rapid response. So if you'd like more details on how we do that, like I said, feel free to email me. I'll throw it in the chat. I'll put my email in the chat if that's okay for the organizers, for folks to get a hold of me.
Please do, Caitlin. It's appreciated.
Yeah, absolutely. Thank you.
Thanks, Kate.
Okay. So the next question from Heather, and I think this materialized while Matt was speaking, but if anybody has any insights on this, this is something I don't know anything about, and I don't know if there's anything to be known about it, frankly. The question is, any thoughts on whether microplastics might be affecting bloom size or bloom toxicity?
This is Brenda Lefrancois. I was a co-author on Matt Hudson's presentation from the National Park Service. And I don't think that we have any indication that microplastics have played a role in the current, observations of blooms in Lake Superior. We have done some work on microplastics in collaboration with UMD's Large Lakes Observatory. So we know that they are out there. We found indications that they're on beaches and in the water column, but concentrations are fairly low. And we haven't seen any relationship with, the locations of where blooms have occurred in the past.
I can't imagine there would be a direct effect, but I could imagine there might be some kind of trophic cascade effect, especially if microplastics are being ingested in favor of some other thing that might have some interaction with blooms. But again, I don't think there's any research out there on that to this point. And again, if any of our presenters have any info to the contrary, I'm really keen to hear that.
This is Hunter. There was actually a paper that came out in the last 6 months in Environmental Science and Technology. Can't remember the name of it, but it was looking at cyanotoxins, I think microcystin specifically, foaming onto microplastics and creating kind of a trophic cascade once that got into the rivers from wastewater treatment plants.
So that in essence, the microplastics are functioning
basically a vector,
attaching to toxins because of their small size?
Yeah, that's the first thing I've seen on that, though. They're just acting as a vector for the microsystems that weren't removed.
So a carrier, in essence, a carrier of toxic chemicals potentially.
Yep.
Interesting. Thanks for that, Hunter. Hannah asks, how are cyanobacteria propagules thought to move or arrive in uplands when blooms primarily happen in lakes?
I can answer this question. So a lot of those, all the propagule stuff is related to work that I did during my PhD. So, one of the first questions that we had when we were looking at blooms in Lake Superior are these blooms actually originating in the lake, or is there some sort of upland connection? And that's what led me to those first experiments, where I took samples from rivers near the harbor and in the lake, and then I brought them back to the lab. So yeah, that's an open question right now, is, are the rivers just kind of this conduit where the cells are coming through from an upland location that might be more productive? And we just don't really know. But as you saw in the presentation that Matt gave, there wasn't this kind of like smoking gun location where we were like, this river is really, has a lot of potential for being a hotspot or a potential, seeding location for blooms. And so a lot of those locations are actually, they're forested. And so we don't have kind of what you see in a lot of other systems where there's this really bad lake, that's really well known for cyanobacterial blooms. For some of those locations, we have seen some surface scums upstream, but not anything that's just like this really crazy looking sludge where you're like, yes, we know exactly where it's coming from. And so kind of what our running hypothesis, and this actually addresses, there were several kind of questions that were tied to this. Kind of what our running hypothesis is that there are low levels of cells in these rivers. So you got to kind of remember the distinction that we're taking these water samples from rivers and lakes. And so the second experiment that I did, it was lakes and ponds, rivers, as well as coastal areas. And it was still rivers that actually came out to be the most likely contributors of seed populations to Lake Superior. But we're collecting these water samples and then we're bringing them back to the lab and exposing them to what I always like to call the yummiest conditions for growth. So we're really trying to promote growth in the lab. So it's not like we're finding these big blooms in these rivers. That's why we really characterize this by potential. They have high potential to be sources because there's not just bloom material chugging down the river and making its way out to Lake Superior. When we did our field observations, we also collected in situ temperature and biophysical and chemical characteristics. And we found that those places that had high potential were characterized by low temperature and high conductivity. And there were as many other parameters that we measured, but those were the ones that were statistically significant. And so these rivers are actually often lower temperature than the near shore of Lake Superior. And so I shared the link to the open access paper for this if you want to take a look at it. So we highlight a lot of the stuff I'm talking about. And so what we think is going on is that there's kind of this really low concentration of cyanobacterial cells hanging out in rivers that may or may not be transported from upstream locations. And then they're making their way out into the near shore where it's actually warmer and there's more light because a lot of these streams are covered by a lot of canopy cover and there's not a whole lot of light coming through. And also it's a lot less turbulent. So they're viable and they're hanging out. They're coming out into the near shore where conditions are more favorable with higher light and temperature and more stable water physics. And so they have this opportunity to bloom, and particularly in those really large storm event years, there's this big flux of nutrients to the lakes, which is probably contributing to those larger events. And so that's kind of what we think is going on right now.
Great, thanks, Caitlin. So Chase asks, Hunter, when using a YSI XO1 chlorophyll A and phycocyanin sensor for monitoring, do you have a threshold for either or both of those parameters when deciding when to trigger further sample collection? And if so, how did you develop those thresholds?
Good question. Not yet. We're still working on that. I've only been using that for about a year, and it seems like we've always got some background chlorophyll A in the waters. But whenever you get a phycocyanin detection, you can pretty much expect to find some cyanobacteria in the samples as well. That seems like that's correlated directly from what we've seen so far. We also did some sampling during a really big bloom where we took samples from the surface, midway down in the lake, and then at the lake floor. And then we ran samples on the flow chem as well. And we saw some pretty direct correlations with those counts too. So that's really more of a flag to say, hey, go collect a sample. You've got some phycocyanin, so you've got some cyanobacteria out there. It's either a go or don't kind of number for us at this point.
See, so it's the phycocyanin that is most obviously related to presence of cyanobacteria blue mite. There's no, you don't have a set number yet. You just
no.
Okay.
No, that's in progress. Hopefully that'll be something that we establish in the next year or two. We just need more data.
Thanks so much for your reply. Anja, I think your question has already been pretty thoroughly addressed. So to not give it too much more time, if it hasn't been thoroughly addressed, please drop a line to Matt or Caitlin, his co-author, if you need more of that addressed than has already been discussed here today. Colin from Dane County Land and Water asks of Heather, and I'm afraid Heather is not with us. So has Heather had to step off because of internet connectivity? Have we lost Heather?
That does seem to be the case.
So assuming so, and just in case any of the others have some experience with this piece of hardware, I'm going to ask the question anyhow. So if you have something to offer, please do. If not, we'll move on rather quickly. Have multispectral cameras proven effective as real-time detectors of cyano-HABs? And could you provide any details or references on the processing workflow? Anybody using multispectral cameras?
I'm not, but I'm going to say yes, because I know that there was a big chunk in the new M57 from AWWA where they're going to discuss that. So yes, but I don't know a whole lot on that.
I'll just add, I'm not using multispectral cameras, but I did use in my experimental work a phytocam, which is using, multispectral fluorescence techniques to differentiate between roughly brown, green, and blue-green algal groups. I don't know if there was a specific question related to that, so I'm happy to follow up with that if needed.
Sure, and do write directly to Heather if you'd like as well for a reply. I will add just a little bit that Lake Erie has some pretty chronic harmful algal bloom issues, and there's a fair amount of remote sensing using satellite imagery and processing that with algorithms specifically to detect phycocyanin fluorescence from satellite images, but that's probably a different scale than the original question is asking. Still, there's a fair amount out there on that kind of post-image processing as a nearly immediate detection, at least as soon as the satellite images become available of the current location of a particular bloom. Jeffrey asks, increased salt levels from winter road treatment runoff do cause stratification for inland lakes. Lake Superior is a very large lake, but accumulation over decades could cause impacts. Has anyone conducted salinity level testing at different depths in Lake Superior to see if there is any increase? I mean, Superior is big and deep, and it certainly has a chemocline, but I don't know the specifics of it.
Yeah, I think that's a really good question. Kate responded to that. We're not aware of any specific efforts that are looking at that in Superior. I do know that Superior has the lowest salinity levels of any of the Great Lakes. So there was a study that came out last year related to salt in Lake Michigan and salinity levels that have been increasing. But as far as that affecting stratification, I think at this point, it's the climate drivers related to earlier onset of stratification that's due in part to less ice cover on the lake in the winter, and that allows the water to just warm up quicker in the summer. And once it stratifies, then that surface water can really warm up. So I think it's more of a climate related driver in Lake Superior than salinity, but that is a good question.
That makes sense to me too. I mean, the chemocline of Lake Superior is where I'd expect salt to accumulate, and I can't imagine that that's often disturbed. Tiffany's asked a rather specific question for Heather, and with Heather not on board, I'm not going to take the time to read the question. Tiffany, if you're listening, please do feel free to write Heather directly. Anthony also asks for Heather, and I don't know if this is a little more generic, and so Matt and Caitlin might have some perspective on this from Superior, but I'm not sure. So Anthony asks, is anyone looking at the HAB effects on native mussels? Mussel communities in general are stressed and declining. Illinois Natural History Survey has a good record of mussels in Illinois. So unionids, have either of you looked at unionids in any degree as related to harmful algal bloom organisms?
I have not. Brenda might actually be a better person to ask for this. I see Brenda's unmuting herself.
Thank you.
Yeah, thanks, Kate. I'm not aware of any work along those lines in Lake Superior, but I work broadly with national parks throughout the region. And one park that I know has paid attention to that is St. Croix National Scenic Riverway, which has a long history of native mussel work. And there was a project some years back that we worked on with US Geological Survey out of La Crosse, looking at effects of really the food quality effects of harmful algal blooms on native mussels. So what we were seeing was this big gradient in food quality, lipid content, those sorts of things associated with harmful algae, and then looking at the effects of that on unionid growth and so forth.
Interesting, but not specifically toxins, mostly food quality.
Right.
Okay, and what were the effects?
Gosh, I'd have to look back at the paper. It's been so many years now, but I think the bottom line was there did look like there were some effects, especially on juvenile mussel growth, not a survival type of impact, but just sort of a food quality effect on growth,
So
especially in early stages.
I assume poorer growth just because cyanobacteria are so lipid poor compared to like a diatom.
Exactly.
Okay. Great. Thank you so much. Yvette, from the South Central Climate Adaptation Science Center at the University of Oklahoma asks for Hunter. The FlowCam seems to be a great way to do preliminary analysis for labs, agencies monitoring for FHABs. How satisfied are you with it? And is it affordable for state agencies on limited budgets?
Well, we love it. It works great for us. I tell everybody, you just have to manage your expectations. It's any piece of equipment. You're not going to press a button. It's not going to run automatically. It's not going to remove the need for QA/QC and just give you answers you're not going to have to review. You can streamline the process to where it runs very smoothly and just about as automatic as possible, but you can't completely remove the human factor from that. As far as affordability, it's pretty expensive, but if you're like us, I can ask for a $40,000 entry-level lab tech for 10 years and won't get it, but I can ask for equipment and instrumentation and that just shows up. So at least where we are, it's easier for us to get new toys than more people. And it's been great for us because it eliminates up to 4 hours of work or more during a bloom, and we can run samples within a couple hours of getting them collected. They get back to the lab and we have answers on what's in the water and can make more real-time decisions.
I'm a big fan of toys, but I'm also a big fan of bigger staff. So yeah. I'm a little torn on your answer, but thanks for it just the same. Claudia asks, Zoe, when monitoring agriculture nitrogen, do you differentiate between liquid manure and chemical fertilizers?
Currently, we don't differentiate is that one of the reasons we selected the cone fraction in the watershed as an input is that we, as the development of the satellite techniques, we can easily get information on crop types there. However, it's really difficult to get accurate data for which type of the fertilizer or which type of the manure the farmers apply and specific field. Needless to say, the accurate data in a certain watershed. Thus, if we could use detectable like the corn type and crop type we can easily get from the satellite data. We can easily scalable our studies into a broader region. And another consideration here is that with the development of satellite remote sensing techniques, we can get more detailed information of managed practice like tillage, cover crop, and in the future we can use once we have the field experiment in several plots, and we develop the empirical relationship between the different practice and the nitrogen loading change, and with observations in a broader scale with satellite images or other remote sensing techniques, we can easily assess the managed practice, how different managed practice would change the nitrogen loading in different watershed.
Thank you, Shui. Matt Gladfelter from Auburn University asks, Hunter, can you tell us more about the water source and the animals present? I was curious if you're aware of any fish that could be suppressing zooplankton, thus promoting both algae and cyanobacteria growth.
It's a really good question, and that's not something we've looked at. Our lakes are extremely turbid. We've got a lot of suspended clay particles in our lakes. You can't see more than a foot down because of the turbidity. So I don't really know how to answer that because we haven't looked at that, but we do have a lot of white bass, a lot of white crappie, catfish, perch. And so I would expect that to some degree. We don't see a lot of zooplankton in the samples we run on the flow cam, unless we use a lower magnification and a wider flow cell.
There's of course quite a bit on phytoplankton in general and trophic effects on, suppressing zooplankton or not by fish, whether or not that results in more phytoplankton in general, but I'm not sure about specifics related to cyanobacteria. It's probably out there. Else is familiar with that literature, feel free to chime in. This question materialized during Heather's talk. Now, Heather's not specified, but it might be something that others can address as well, especially those who are sticking probes in the water. So I'll ask it, and if it can't be answered, we'll move on if it's too specific for Heather's particular suite of hardware. What specific phosphorus sensor is attached to your fixed super sensor? So don't know if any of you are using similar hardware, but if so, please offer up. Hearing nobody clamoring to reply, let's get to Tiffany's question, who asks, Hunter, do you notice any effects on submerged aquatic vegetation or animal species in the lake when you treat with copper sulfate?
No, not really. Again, same kind of thing. Our lakes are so turbid, you can't see to the bottom of the lake, even at the boat ramp. But we dose with 1 part per million copper sulfate, so we don't have a fish kill or we don't have negative effect on the aquatic plants. And it's further down on the question list, but I saw somebody ask about permitting. In Texas, we're not required to have permits to apply that to the lake because of the type of copper sulfate we're doing. I guess it's already approved by the state for water treatment.
Right. Now, copper sulfate is, of course, a really commonly applied algicide, the universal algicide, right? And if there are effects, it's probably residual in accumulation in the sediment over time, but those are often buried by new accumulation of sediment as well. So there's a fair amount of literature out there. Most of it's going to be related to residual effects in sediment. I'm not sure. Let's see. Atefa asks of Heather what satellite data she's using. Now, I don't know if any of you are processing satellite data, but if so, please feel free to offer up what you are using. If not, and if you're not familiar with Lake Erie's harmful, I should say cyanobacteria forecast system, that's run by a guy named Richard Stumpf with NOAA. Look at that because he does a fair amount with satellite image. And that might be of use to you. Drop me a line if you need some contact information. But if any of the rest of you are using satellite images, since Heather's no longer with us, please chime in on satellite images. So Jesse from the Minnesota Pollution Control Agency asks, regarding the rivers feeding BGs, I'm guessing that's blue-greens, to the South Shore, and I'm guessing the South Shore of Superior, are there eutrophic lakes in the headwaters of these streams that may be the source?
Kate, do you want to take that one? You kind of addressed it a little bit earlier.
Yeah, can you repeat it? I had to leave and rejoin, so I don't have the questions in the same order.
Sure. So regarding the rivers feeding blue-greens to the South Shore of Superior, are there eutrophic lakes in the headwaters of these streams that may be the source?
There aren't any. Yes, I touched on this a little bit before. Not really that we're aware of. We're not like, okay, here is a big problem area. And I think I saw a few other questions that were asking kind of like maybe land use related type things. And so as Matt mentioned in the presentation, this Lake Superior is very interesting. So it's cold, it's oligotrophic, so it's low nutrients, or it's relatively cold. And it's not a highly developed area. So the most highly developed area in the Lake Superior watershed is actually the Duluth Superior Twin Forts area. And that's even still like not nearly as developed as a lot of the other areas where we see big cyanobacterial blooms in the Great Lakes. And so Lake Superior is really kind of unique in this situation. And so with Lake Superior, we think it's more so not driven by like these, I guess, problem areas, I guess you could say, on the landscape, but there's a lot more of this connection to climate factors, like these major storm events, rising temperatures. So like while Lake Superior is cold, it's one of the most rapidly warming lakes in the world by summer surface temperatures, and it's the most rapidly warming of the Great Lakes. And so while it's relatively cold, it is warming, and the species that we see in Lake Superior, Dilacospermum levermanii, actually has a really wide range of optimal temperatures from 13 degrees Celsius up to around 25 or so. And so we also have this species that is maybe more adapted to these types of temperatures. I think Matt also mentioned that, you know, this species has been implicated in blooms in some of the Alps lakes and things like that. And so it's complex, but no, it's not the same story that we see in a lot of other HAB situations where it's like it's high temperature and it's high nutrients, where, you know, it's a very different system. And so we're very much in the process of kind of sussing that out right now.
Great, thanks, Kate. Maxwell asks, hi, Matt, any data on how long these blooms have lasted once observed in the lake? If they're seeded in rivers, do you expect them to die off quickly in the cold and nutrient poor lake environment?
I'm also going to grab that one, Matt, because that's related to the stuff. So I don't know if the person who wrote this question earlier had heard what I spoke about previously, but the lake is actually warmer than a lot of these rivers. So a lot of these rivers are 10 to 15 degrees max, you know, during the warmest parts of the summer, whereas the lake, the near shore of the lake in particular, could be anywhere between 15 and maybe 23 degrees Celsius. And so oftentimes, especially the surface waters of Lake Superior are warmer than a lot of these rivers, where the water is constantly in motion. There's a lot of canopy cover. You know, it's kind of backwards from what you would think. And so we actually have data that show indeed that the nearshore is warmer. And while Lake Superior is nutrient poor, it's actually very, very high in nitrate. It's a really high N to P ratio. And then the rivers are low N to P. And so one of the things we've kind of wondered about is right at that interface of where rivers hit the lake, is there kind of this really nice mixture, like a stoichiometric mixture of nutrients. And then in these really big storm years, you're getting big fluxes of nutrients into the lake from all this sediment erosion. And so while Lake Superior generally, yes, is nutrient poor, there might be these kind of short-term bursts of nutrients that really kind of jumpstart these really big blooms. But we have characterized blooms in the past as minor blooms and major blooms. And typically we see minor blooms in years where there's no storm event, so there's not this big nutrient influx, but there is higher temperatures. Or there are higher temperatures, and then these really big blooms like we saw in 2012 and 2018, they have both higher temperatures as well as these really big storm events that are delivering large amounts of nutrients. And so a little bit of this is not really, it's a little anti-intuitive from what you would expect for Lake Superior, which also makes it very interesting.
Great, thanks, Kate.
Yeah, and Kate, I'll just add to that, you know, like the temperatures in Lake Superior, you vary widely. So, in the week before our big 2018 bloom, we had very cold temperatures due to upwelling, temperatures around 6 centigrade. But those were followed by, nice warm temperatures. So that when these cooler trib waters meet the lake, they are greeted by warmer, clearer temperatures, like Kate was saying.
I once did some brook trout work in the Porcupine Mountains and living from a tent for a while, and I bathed in Lake Superior. From the perspective of bathing, Lake Superior is always cold.
You get used to it.
True.
Thanks.
And I
We
think,
probably go ahead.
Oh, I was just going to add that the first part of that question, I think, was about how long the blooms last. And I think we should note, based on that, and maybe one other question that was in the queue somewhere, that these have been really short-lived events. Even our longest-lived events was very patchy and, lasted probably a week or so. So, relative to a situation like the Lake Erie blooms, we're still at very early stages here, and things do tend to dissipate quickly because of lake circulation and other factors.
Thanks, Brenda. The people of Lake Erie are envious of you. We'll probably only get to one more question before we break for lunch. So I'm going to ask Heaven Lucero's question. What is the process like to test for these algal blooms? Would it be as simple as a grab sample being sent to the lab? And I imagine everybody who has some experience of the morning session could toss in some thoughts.
I'll start. I'll say absolutely. That's where you're going to start, surface sampling, but you have to be careful because the bloom might be a foot down, 2 feet down, 5 feet down, depending on the water clarity, temperature, and other parameters. So you have to do some investigation to see where the issue is and make sure you get a good representation when you collect your samples. So you can use something like a Van Dorn sampler or other kind of sampler to collect samples at different depths. But grab samples are normally what we do.
Great, thank you. Any of our Lake Superior folks care to weigh in? Or do you feel like you've already addressed it with our previous questions?
Yeah, I mean, that's pretty much step one, is just collect a sample and pop it under the scope and see who's there. And then we pretty routinely will preserve a sample and then also send it out for toxin analysis with Todd Miller at UW Milwaukee. Brenda or Matt, feel free to add to that.
Yeah, that's about it.
I think you're spot on.
Great, thanks all. Personally, I've very sincerely enjoyed the morning session. I think our presentations have been excellent and provided me with a tremendous amount of information to think about. And we're going to break for lunch. So before we do, I want to once again ask any of the speakers who are around. There are a lot of questions we simply couldn't get to. So if you have a moment to look over the questions, and if you have a reply, if you have a moment to type that reply, it's appreciated. If you don't have that moment, it's certainly understood. And I thank you for your presentation as well anyhow. So thank you all for being here. Thank you to our speakers. Thank you to our attendees. And thank you for providing such thoughtful, interactive questions to make this discussion panel to follow worthwhile. And again, thank you especially to our sponsors. We are to break for lunch, and we will begin the afternoon session promptly at 12:45 Central. Thank you so much for attending. Anne, Amy, do you have anything to add before we break?
No, I see that Anne dropped the speakers' email addresses in the chat, so feel free to reach out to them. And I have also put our website in there where the recording should be available early next week. And look forward to seeing you again at 12:45 today.
Thanks so much, everybody. I look forward to it as well.